back in our lives. Future Cannabis Project FCPO2. Thanks everybody in chat for joining us, and everybody on the replay who's going to rewatch this one, myself included. That's kind of weird saying thank you to yourself, but hey, I just did, so we're rolling with it. It's Friday night, and we're back with SunGrownMidz talking some genomics and cannabis plant breeding. Thank you for joining us again, Trevor. I appreciate that.
Of course, Jed. Thanks for having me on, man. I appreciate the invite.
Yeah, no problem. My pleasure. I wish, it's kind of funny. We're having a great conversation beforehand. This is going to be an excellent, addition to where we were going. Um, it's funny. You always have some great conversations before the cameras start, don't you? But so tonight we're going to be talking about a few things that from the breeder perspective, the cannabis breeder perspective, are going to be important. These are usually traits or characteristics such as like yields or secondary metabolites, things that are important to a person looking to breed the next best cannabis cultivar. And so this one is definitely going to have, a very close application to what a lot of people are doing. And it was funny that we were talking earlier beforehand about like cannabis breeding books. And, I've got a few of them and I've read them and they're great. There's a lot of information in there, but there tends to be not a complete knowledge set, and it's not for lack of effort or anything. It was just kind of the information that we had at the time of when you're doing these things almost anecdotally, just kind of based off perception. You get a certain rule set, and a lot of this rule set, like I said, is it's kind of like cannabis breeding. This is how you breed cannabis, but then there's also the whole plant breeding world. And I love that you help bridge the gap between the two, because it's really picking up where that cannabis knowledge has kind of left off. And this information is actually what's going to get us a lot further down the road. So thank you once again for coming back and dropping some of this knowledge on us, man.
Of course, yeah, no, thank you. And I think, yeah, that's a good lead in. I feel like to a certain extent, some of this stuff, there's been a hesitancy in the cannabis publishing world to dive into this level of information, just because when you start getting into quantitative inheritance and heritability and these sorts of things, to actually read all the literature, it's incredibly complicated math and very hard to understand. We're going to break down some of it and go into some of the different statistics and stuff. And I tried to keep it as light as I could. I'm certainly no statistician myself, so
Thank you.
Tried to keep it light on the math. But like that's it's something that Tony said a couple weeks ago. I think it was during the seed collectors discussion in chat. He mentioned that the breeding is math, and that's when you get down to it, you're dealing with frequencies and statistics and a lot of variance that you're trying to figure out without necessarily knowing exactly what you're doing. And we came up with these mathematical systems or theories for approximating what was going on. And I hope tonight we get to kind of go through the theoretical mathematics, but then bridge that gap to some of the genomics and how this stuff's being applied today, and like really kind of the difference that, we talked about the calculator and the pen and paper sort of technique. We're going to do that heavy on the first half, and then on the back half, we're going to talk about how you send it to a lab and the lab tells you whether or not something's there, or, those sorts of marker-based analysis.
And it's good to know their methods. It's good to, almost be able to, you don't have the same equipment, so you can't necessarily double check their work per se, but you can tell if what they're giving you is kind of making sense, at least by having a foundation. And
So
one of the things I kind of actually had written down from the last time we talked, speaking of math, I love math. Music is math as well. Music has been my life. So yes. But I had a question here. It's like, how close to an algorithm for awesome weed are we with all that we know already, just by doing the math? Like, is there a algorithm for typing the fruity awesomeness? Are we getting there?
um, we're getting closer. Like, there's definitely. I wasn't able to pull it up just this evening, but you can see kind of the older maps. The like we talked about linkage maps before, and linkage maps are kind of, if you were to have an algorithm, you'd need a linkage map to know like this gene's on this chromosome here, et cetera, and like build out the whole linkage map. And they've done this with like fruit flies
Mm-hmm.
and mice and model organisms. They've mapped the entire genome of these things, not just through sequencing, but through traditional linkage map assemblies.
Wow.
And yeah, and like it took decades of work to do this, but they did it. And essentially you can see the early rudimentary steps that were happening about 20 years ago in the linkage maps, and it was very sparse. And then you can see what's going on now, and even some of the stuff now is pretty sparse, but it's getting more and more, they're filling out the chromosomes and finding where the different genes are and what their associations are, so that a lot of the stuff that we're going to talk about in terms of quantitative inheritance and traits have been figured out through more advanced genomics. And that's really, it's going to save decades in terms of the actual time and labor to figure this stuff out.
Okay. And that kind of makes me wonder because, science does rapidly advance and sometimes we're building models off of something this big and all of a sudden we understand the thing that's this big and it changes our perspective. Does that come into play at all with plant genomics or do we pretty much have a good idea of what the actual building blocks are?
Yes and no. So epigenomics are still very much understudied and poorly understood in pretty much everything, but in cannabis in particular. Um, that's an area that a lot of people will point to, but really the bigger issues and what's coming online now are um, tran- uh, the transcriptome. So you've got the genome and then you've got the transcriptome. So the transcriptome is what essentially translates the genes into the different proteins that they end up
Okay.
becoming. So this is
Okay.
like RNA and stuff like this.
I
And
understand
so,
that.
yeah. And like when we get into like uh, terpene synthase genes,
Hmm.
they will have a particular process that they do, but they share that same metabolic pathway and process with cannabinoids. And so there's a lot of crossover here. And on top of that, these same gene families are shared by almost every species. So all the other plants that have terpenes have these families of genes called terpene synthase genes. And so we we're going to be able to go through and look at some of those, how they interact with each other, and then how also they interact with cannabinoids, because it's a complex, it's one of those things where we understand that the transcriptome will make the cells do particular things. And so it'll be like, hey, you're going to do CBD or you're going to do THC, and both of those rely on the precursor CBG. So like every one of these is kind of like a molecular chain or link in a chain, so to speak.
Okay.
And so the transcriptomes like complicates things. Then there's metabolics, basically, looking at all the ways that plants metabolize things. Um, There's proteomics. Like there's a whole list of omics that are on top of genomics that
Okay.
are complicating the picture of things. And we'll get to that at the very end. In particular, there's a paper that I just read that it talks about cannabinoid inheritance, and it's it's wild. Like it's so much more complicated than just genes or dominant recessive. It's mitochondrial DNA, chloroplast DNA, all sorts of stuff going on.
That's nature being nature, saying you might get close, but you're not going to figure us out.
Yeah, yeah.
But one thing you did mention, especially with the terpene synthase, and we did have a question on that on my Instagram, and we get to that later. You'll probably actually cover it by the looks of things. But the big thing there I heard is, other plants do create terpenes. It's a universal, almost location, mode of action to create these. So we don't have to wait for cannabis to be widely studied to fully understand this. Like we can actually already get there just by using other plants at least, or get closer to an understanding. Because again, there's always probably going to be some variable that might be different in there.
Yeah, no, totally. That's something that if you read the papers, a lot of times when they're looking at and doing like early research into quantitative traits, they'll rely on the same trait structure or the same trait in a different plant. And so for terpene synthase genes, they were able to identify and locate a lot of them because of their similarity and their shared structure with these other terpene synthase genes that are going on in other plants. There's also a paper that's about the genetic mechanisms controlling flowering time, and same thing, where there's a set of genes that are associated with those traits, and they've already, completely mapped them out and know what they are in cotton, maize, rice, a whole bunch of other crops. And so they've been able to find the same set of subfamily of genes in cannabis, and it's helping them to elucidate the flowering time mechanisms.
Wow, and that's huge too, and that has a big commercial application. So that's something that I imagine is going to be looked into a lot further with R&D dollars from a lot of these big companies. The faster they can bring in a harvest, the better they're doing. So that is, follow the money. That's of interest. So I'm sure that'll pop up at some point.
Totally, and that's it's really interesting, and this is something that Camara, he brought it up, and there, if you look at the early research into cannabis, so much of it is determined by the research agendas of the different agencies or entities that are funding the research. And all of them in the United States had to have DEA approval. You've got the University of Mississippi and all the research that they did. And pretty much most chemotype information, the early studies establishing standardized protocols for doing GC and all the different chromatography, as well as a lot of the early genomic studies, were really oriented towards a forensics end goal. So it was all about fingerprinting strains, varieties, and identifying and disrupting drug trafficking networks. Like that was the whole rationale for the research for probably the first 30, 40 years, from like the 70s. 69 is when University of Mississippi started operating. So from like 69 to the present.
And they grow some pretty bad cannabis down there in Mississippi. But to your point, to the fingerprinting, the kind of the practical application of that was, let's say Joe Bob is just growing, he's the original. This is completely fictitious story. People get sensitive sometimes, but let's just say Joe Bob in the hills of Minnesota is even wilder. Came up with the original sour D. If he has the original cut, it has the original profile to it. Now, if he were to be busted and they were to fingerprint his cannabis, well, when somebody gets busted with 20 pounds down in California and it has this exact same fingerprint, that was kind of the practical application of why they were interested. So they did a little bit of our work for us, but it was for kind of jerky reasons, essentially.
Totally, totally. And because that research had already been done, a lot of that was some of the early commercial applications that you were seeing. Like Steep Hill was like
Yep.
strain fingerprinting sort of thing, where you could bring your strain or your variety in, they do the testing and then say, okay, it's got this chemical profile, which is its fingerprint sort of thing, which wasn't particularly successful, mostly because, like, you know, they were dealing with much different populations and sets of variables. So, yeah, comparing like geographical land races that had diverged over many, many years and creating your data sets off of these and saying, like, because this is what Carlton Tucker did. He was the drug czar under Reagan. Yeah, he doing research into him. It's incredible how successful and dumb people can be sometimes. And like, the things he said were just mind-boggling. He claimed that smoking marijuana increased your propensity for homosexuality, and that's kind of been characterized for why he lost his job.
Okay, yeah.
Yeah.
I totally remember quotes like that and people like that. Another one that comes to my mind, it was after Washington and Colorado went, but Utah was thinking of medical, and there was a sheriff. They were doing this bust out in the woods, and they were talking about these stoned rabbits. And these rabbits had lost fear of humans because they were still trying to come around and eat the leaves while they were destroying it, and it was going to be an ecological disaster. And yet, dude, it's like searchable and findable on the YouTube's. But yeah, how can you say these things with a straight face? Don't you have like, didn't you go to college, man? But
Yeah.
yeah. Yeah,
No,
so I get your point, but Reagan really was when the drug war kicked in for a lot of people. So this person was highly effective at their job.
oh, fully. And like, he saw himself as like, he was an evangelist for the drug war. And yeah, so it's interesting going through that history, you know, because he was one of the people who advocated spraying paraquat on cannabis in Mexico, and like argued that the known health effects of paraquat consumption were less harmful than the like scaremongering he was doing about the health effects for cannabis. So he's a wild figure, and there's a whole host of other folks that continue. There's like New Haven, Connecticut has a whole research department dedicated to it. It's a very interesting story, the history of academic research in cannabis, and I feel like it was a really shitty story until about like the last 10 years. And in the last 10 years, it's not just people who were doing it because they had government jobs and were being paid to essentially like perform a task, which was usually in furtherance of prohibition. It's people who actually care about the plant and are interested in doing the research and developing it for our sort of uses.
And growing it proper too. Again, you mentioned Mississippi University there, and what they're growing isn't even like street level cannabis, consumer level cannabis. You can't, it's like comparing apples and oranges almost from what I've seen there. And how I've originally understood it too is, a lot of these studies were done by NIDA, the National Institute of Drug Agency, or something to that effect. But NIDA was really only interesting in funding things that were looking for a negative.
Yeah.
They weren't going after any positives. They didn't want to hear the positives. So a lot of the research, kind of like you're saying, is skewed and to the genomics kind of breeder level, probably not undertaken in a serious like cultivation manner, more as you said in a law enforcement manner.
Mhm.
I find it completely fascinating. Well, all this stuff's fascinating. But you mentioned that, you know, like flowering time was located, and that's kind of universal. Terpene synthase is kind of universal, and we're able to study those in other plants. And I'm not into, you know, the GMO aspect of things, although I know we're going to see it, and I know it's going to create better cannabis. But there's things, you know, like we put salmon genes into fruits to preserve color. Um, the one thing I always say about like cannabis and the benefit of studying it like this is we might find something, even if you're completely against cannabis, we might learn something through studying cannabis that is going to affect you, and you can go derive your pill form of that, and that's fine. And a good example is the endocannabinoid system. We didn't know about this a long time ago, and we really kind of found it through cannabis research, but it seems to control a lot of things in the body. So even if somebody is anti-cannabis, I still think they need to support the study of it, and that's, I don't know, a long way of kind of agreeing with you. But you know, we never know what we're gonna find, and we need to dive deeper. So I appreciate you indulging me on that little rant there, but that's let's see here. GMO will improve cannabis. How do I think GMO will improve cannabis? I see that one pop up, and I'll just touch that real quick. The IPM, being able to grow it in certain areas. There's a lot of things you can do to a plant, and I'm not rooting for it, but I see where it makes sense for certain applications, and I'm probably just digging myself a hole here. But I saw that pop up in chat, and I just wanted to address it because I put it out there. Yes, you know, we probably will see it, and yes, it probably will make some awesome stuff. But I'm gonna go for SunGrownMidz packs and continue to grow that stuff because I know you know your business.
Well, thanks. Yeah, no, I'm not like, yeah, I've been very anti-GMO. I've been very anti-CRISPR. I
Yeah.
think those things are inevitably going to happen. Um,
The
it's
money,
just,
it has to.
yeah, and
Sorry to interrupt, but it's you know, again, I hate to be that devil's advocate, but it is going to be an industry, it is going to be business, and it is going to be about money. Doesn't mean it
yeah,
has to be for us, and that doesn't mean it has to be for everybody. We have a good community. I think we're going to be fine. I think we're going to be better because of it. But to deny that something like that's coming, mm,
yeah,
sorry to interrupt you there.
yeah. No,
I get emotional.
I fully agree that it's going to happen. And there are going to be people who will argue and debate over whether or not it's a good, like, specific application.
Right.
Um, and then there's just going to be people who are like, flat out no. Immediately no.
No.
So,
No.
and I understand exactly. I fully understand that. I think that there's a lot of cautionary tales there. There's a lot of reasons for hesitancy and skepticism. It's a market strategy that will be deployed by people who are operating in the cannabis space
Yeah.
in a way that gives zero fucks about what we care. And
Yeah.
so,
Yeah.
you know, it's going to happen. I think that it remains to be seen whether or not, like, just because I remain skeptical just because of the Roundup Ready sort of like scenario where, like, by and large, I mean, okay, I grew up as a kid in like the 90s, and the alternative globalization or anti-globalization movement,
Yep.
like the WTO,
Yep.
and like had all these posters from there of like that rally that were anti-GMO, like that was one of the main talking points that was going on in that time period. And we were told how it was going to revolutionize things and it was going to make these great results. We haven't really seen that. We've seen Roundup Ready crops adopted, and that's, you know, increased pesticide applications, but that hasn't really done all that much in terms of overall yield improvement. It's certainly not like Butang and like traditional breeding, or Bulang, sorry, with his with the breeding of the dwarf wheat varieties that basically created the Green Revolution. Like those definitely had some like negative consequences and side effects in terms of intensive agricultural sort of processes being exported all over the world. It also had massive yield increases that have helped feed millions of people around the world. So like both of those two things are true. That isn't, you know, we haven't seen those similar trend lines with GMOs relative to yields and trait inheritance gains necessarily. But the one area it has been applicable, and we can get off the GMO topic. When I went to UC Davis, like they start the discussion when you talk about resistance breeding at GMOs at this point, because in so many crops there have been just, you know, massive like complete wipeout of crops has been either predicted or imminent from different pathogens. And then they've done some sort of like papaya as an example, bananas are an example, oranges are an example. So like these are instances where essentially to address catastrophic crop failure, they've introduced GMO crops supposedly to like address that issue specifically, and there just wasn't another solution. And so the dude literally posed the question, and like these guys were true believers on GMO. They were like, you know, like talking evangelically about how like, well, this is going to get people to buy into the shit.
So no, that level.
yeah. And but the thing was like, do you want your morning to start with orange juice or not? Was like essentially how they felt GMO technology was going to be normalized, because Americans want bananas, they want orange juice, they want these like staple foods. And if crop failure or GMO is kind of the dichotomy they're presented with, they'll choose that. But we're not at a point where we bottleneck the germplasm so much, and we haven't even really even scratched the surface or begun to investigate this specific area of resistance traits. Like there's a little bit, but not much. Like we're not there yet. And so we like having these discussions with them. We're like, yeah, but what do you do before you get there?
Right. It'll be interesting to see if they come back to kind of revisit that issue, just kind of like banana collapse. That was one that I was kind of a little familiar with. But instead of just, you know, band-aiding it with a GMO fix, if you could actually go back and breed for it. And, you know, it's a long route. Again, it's almost kind of like a cannabis breeder stabilizing a line versus making an F1 and putting it on Instagram. So
exactly. That's, I think, you know, somebody mentioned hearing professors talking about the same approach and in the same way. And I think like a lot of it has to do with like, it's in fashion, it's high tech, it's something that's cutting edge in the field that makes you relevant. Whereas like going back and just like hunkering down and trying to improve resistance through traditional breeding methods is like, it's not only labor and time consuming, it's just not like, it's not sexy.
Yeah, and I didn't fully appreciate that fact until I was in my school program with WSU. They were it's at market now, but the Cosmic Crisp apples. We were finishing up
Mhm.
field trials for that, and talking with one of the professors, Gary Molden. We had all these seeds, and I'm like, so I try to approach him like cannabis. I'm like, yo, you ever like plant seeds and like what do you get? Look for like different phenotypes, and he just looked at me like, that's not how we do it. and I understand now, but yeah, I
Yeah.
understand why. To kind of to your point, it takes a long time. It takes a long time to do that, and
That project was really impressive, and that's something that is a really good model. Now, granted, apple breeding is a whole different beast than cannabis breeding, but that was like a farmer-driven sort of operation where it was a. I've read a little bit about it being like a participatory breeding program where the farmers and the professors worked together to develop an agronomically superior apple, and supposedly it's Kronk as well. I haven't had it.
it's good. It's in the market. I think it was exclusive to like Washington for a year. But also the back end of that entire experiment, how they licensed the apple and they controlled the distribution, is a study in itself.
Mm-hmm,
So
mm-hmm.
that's fascinating. And again, that can relate back to cannabis in a way if you want it to. But that's off of our beaten path for this evening. So yeah, Cosmos Crisp though.
Cool.
So
Well, yeah,
yeah, I know we got I know I'll keep my eye on chat. So chat, if you guys have some questions, definitely get them in, and I'll try to get some up towards the end of the presentation here. But hey, if you caught our first round, you know that we've got some awesome information, and I just want to thank Trevor ahead of time again for putting time into building these presentations. I'm sure you guys all know and can appreciate it, but making these things do take time. You got to compile, and I assume you're like me. It's like accuracy is important to the point to where you beat yourself up to make sure like every line and every sentence is as accurate as you can possibly verify. So I do appreciate the time you've put into them, and I'm looking forward to what we've got for this evening as well.
yeah. This one's a lot more copy and paste. So hopefully my professors and all my sources are on their game.
Eh, You got to hang out with a good crowd. Yeah, don't. Yeah, you got to know who to copy paste.
Yeah, exactly. Um, so yeah, I will click into the presentation, and like I said, feel free to interrupt me. And if chat, I can't really see you when I'm doing this, but like, yeah.
Yeah, I got you. I got you. And I guess one question before while you are launching this, that kind of came up is a lot of these models that we're looking at, how true does it hold to like the polyhybrid and then like the inbred line? Are the rules or the is the math kind of the same, or is there an assumption with this that we're starting with a true breeding or an inbred line?
I'll try to uh, where that's relevant. I'll try to
Okay.
talk about that. I actually left out the little subsection on covariance and really Seawall Wright's ideas about the inbreeding coefficient, mostly because um, it's theoretically it's incredible. It's a very awesome idea. It's not necessarily like it's a theoretical abstraction. It's not necessarily a an accurate depiction of a population. So there's that, and the other reason I left it out is just because we cover a lot of fucking math. But beyond that, I will say yes, there's a difference. it's a totally different dynamic than if you have an inbred population. I think, yeah, I'm not to the extent that, like I said, if it comes up in the discussion, I will I'll get into it specifically, but I will say that the main thing with an inbred, and this is something that I forgot to mention last time, is that the reason for inbreeding in Allard, it was in the slide where I talked about some of the ideas that Robert Allard talks about, and he talks about prepotency, and prepotency is when you increase homozygosity at a particular allele for whatever trait. So essentially, when you're inbreeding, what you're doing is you're increasing the rate of homozygosity by 50% every generation. So,
Okay.
so half of, you're either going to get half of them to be a homozygous, quote unquote, dominant, or a homozygous, quote unquote, recessive, and then 50% of the population will still be, excuse me, it's 25, 25, 50. That's how it breaks down. So it's a 1-2-1 breakdown. Each time you self and you do this, you're going to increase the overall inbreeding coefficient by 50%. So it goes from 50% in the first generation when you have a hybrid to then being a 75% in the next generation, 80 something in the next, 90 something in the next, so on and so forth. That's the basic principle on the inbreeding coefficient. Once you reach homozygosity, what you've done is you've reduced the amount of variation that you're going to transmit from one generation to the next, because at that particular allele, the plant can only pass on one possible one possible allele or zygote. So does that answer the inbreeding question?
Yes, yeah, that does. And just the reason why I asked and brought that up is just again for some more of just like practical application. I know a lot of people today when they're putting two of their favorite cultivars together, they might not be starting with something that's been inbred to the point to where it's getting multiple pure lines out of it. There's still kind of the variety. So I always like to ask where that fits into the relationship. And, I appreciate, like you said, you'll kind of mention it when it applies. But yeah, I'm always trying to find that practical application aspect.
Yeah. And we have a really interesting, so the population's been skewed intensely because we bred it for particular traits over time. So it's out of equilibrium, but it's also incredibly heterozygous because it's such a it's one and outcrossing species. So it starts with a high level of heterozygosity. But then when we just polyhybridize things for 30, 40 years, all we're doing is increasing that heterozygosity within the population by adding something into the mix instead of breeding down. And the few people that have kind of done that work, breeding down, DJ Short gets mentioned, David Watson or Skunk Man Sam, different people who have done, other folks more recently, like Tony Greens, he's done a lot of back cross work, going pretty deep. But most people are just doing a combination of different assorted breeding, and it's all based off of phenotypic selection and what's the new thing that gets introduced every year or 5.
Right. Okay. Awesome. Thank you for that.
Sure. So yeah, and again, I can't really see things. I don't know if you guys can see things. Is the
Let's
presentation
see.
up?
We don't have the presentation yet, no. So if you go to, let's see, if you go to share screen, I believe.
Oh, right.
And then while we're doing that, I'm just going to put up a super fun flyer for tonight. Boom. It's like we're hanging out with Charlie Sheen. What's up, Charlie? How you doing? I love your breakdown. Very public, very epic. Winning. Still using that word. I'm sure the kids know what that one means. I mean, what was she doing with a shrimp fork in her purse anyways? I don't know. Something about tiger's blood and Adonis DNA in there. But let's see here. Okay, so you've got your presentation there. If you go full screen, I will add it to the stream here and get rid of our awesome flyer for the night. And then there we go. We got you up, and I will make that a full screen. Cool. So you're good to go, sir. Thank you.
Yes, and I have also a second, so I can see on that. Cool. So quick review. This is what we went over last time. We talked about Mendel, simple inheritance. We talked about Wilhelm Johansen, pure line theory, genotypes, phenotypes. He's also the person who came up with the idea of the genotype-environment interaction. We talked about quantitative traits and the individuals who confirmed that should say confirmed, not conform, confirmed that Mendelian inheritance, quantitative traits conform to Mendelian inheritance. Nielsen L. did it looking at wheat, the color in wheat, and E.M. East looked at the length of tobacco flowers. Then we looked at heterosis and transgressive segregation. We talked about chromosome theory and linkage maps, then population genetics and the different theoretical mathematics that really kind of are going to be the focus of a lot of what we talk about today. Then we talked about plant reproduction and different breeding methods. So that was last time. If you missed it, you can go back and watch it. There's also, you can hit me up for a folder. I have the presentation and all the different source material that I used last time. This is a slide from last time's presentation. It talks about qualitative versus quantitative traits, and this kind of demonstrates the difference between simple inheritance that Mendel looked at on the left side over here versus what we're going to be looking at over here, quantitative traits. So quantitative traits are controlled by the interaction of multiple genes, each with a small effect on the overall trait expression. The quantitative trait exhibits continuous variation within a population. So if you look at height in people or terpene content in cannabis or cannabinoid content in cannabis, those are all, they show continuous variation throughout the population. This is associated with the idea of additive and non-additive variants. So additive variants, instead of thinking about things in terms of dominant recessive, even though there can be a dominance effect at the allelic level, when you're dealing with quantitative traits, you have a whole bunch of genes, and each one of those genes can be either homozygous or heterozygous for whatever particular allele. Each of those is inherited individually, and all of them combine to then to create the expression of the trait. And all of those genes then have interactions with their environment. So each one of those interactions creates the possibility of added variants. They also create the possibility for additional genetic variants. This can be through the dominance effect or through just an additional allele for a trait. Or in cannabis, there's this thing called copy number variants. There are a lot of copy number variants for cannabinoid synthase in cannabis. So the genes that control the expression of particular cannabinoids, particularly THC, have a lot of different copy number variants, which means there's just duplications of these genes. So that's additive variants. Non-additive variants within a population is this idea of epistasis. And epistasis is the idea that different alleles, even though they may be the same, can have an effect just through their interaction with each other. It's a complicated subset of uh, quantitative inheritance. So yeah.
I'm playing around here with the screens. I'm sorry. By chance, sorry to interrupt you. By chance, do you have headphones or anything that you can plug in there? I'm starting to get a little bit of a feedback. It's not your cell phone. The cell phone's muted, but I think I'm just
Yeah.
hearing a little echo back.
Here, let me go just a minute.
Yeah, no, sorry to sorry to interrupt you there,
No
but
worries.
figure
Yeah,
we'll
let me go find a set of headphones.
cool. Yeah, no problem. Well, oh gosh, that's horrible. Okay, there we go. As you can see, I'm moving my room around, guys. I'm getting some new tents. I'm getting some new gear. So I have one of those grower problems of where do I put things now? And I'm playing that shift around the room, but nobody cares. I'm sorry, guys. I just went off on that. This is fun. I've had the pleasure of looking over what we're going to be going through tonight, and I've taken my own notes. So definitely a lot of note taking is going to be going on. I did see some questions in here. Elka, I saw you throw one up there. Thanks for joining us. And we're going to get to that one a little bit later. Also, there was right below it, oh gosh, a question about fem seeds. Where did we go? Okay, there's your bottlenecking. Gary Garden, is it possible for a fem seed grown plant to throw out bananas in flower if excessive nitrogen is fed? I will say absolutely yes. Feeding can be a stressor, and that is something that can just push a plant over the edge. So as far as stress, yes, we definitely can. And Jeff, my man, thank you for caring. I see that there. I appreciate you. All right, SunGrownMidz is coming back to save me because I could talk about some of this stuff, but no, we've got Trevor here to do that tonight. So let's see here. Almost coming back. There we go. I'll put this up. I feel less self-conscious there. Getting back to the quantitative genetics. My lady is not down with another tent. Oh, I'm sorry, Jeremy. Yep, I hear you. I'm it's one of those things. As long as it stays in here, then it's not her problem. But when everything starts spilling in the hallway, it will be. Okay, and we've got you back with us here. So cool. I will throw up the presentation. Can you hear me all right? Is everything sounding good to you?
I can. Can you mute my computer?
Let's see. I just tried to. Yep. Okay, you're muted now. So I muted the computer. And you're going for that. So let's see, to not get a big feedback loop, I'm going to do that. And let's see here. Heather Wright says, what's in your tent? Right now, I am finishing up. I've got one of the Westport's grape juices in there. I've got a couple freak shows in there. Got Burmese Kush, which is the first time running it. Strawberry banana smoothie, first time running it. And a diesel glue, first time running that one as well. So that is what's in the tent right now. I'm getting excited. Those are about to come down. Burmese Kush made the cut. I cloned that one. It's looking great, and it looks like it has some potential. So that one got cloned. Westport grape juice, obviously, that's we're running that one hard. So that's sticking up too. But the other ones, I have more seeds. I'm just kind of burning through seeds right now to find something that I might want to work with later. But all right, we've got you back. Do you want me to, or you got your phone unmuted there? Okay, so yeah, you should be good to go now. Well, let's see here. Not hearing you yet. It looks like it's unmuted from that end. Let's see. This is the Future Cannabis Project. We do it live. Every time. Let's see here. I can't hear you over the phone one, even though it looks like the icon's unmuted, which is kind of interesting. I see the computer's muted. That one should be picking it up. T. Barrington asking, is he on Bluetooth? I don't know. Yes, Ian, you are just in time for the tech difficulties. I remember a conversation a long time ago when I first started doing some of these shows. I remember a comment somebody just typed in chat. It's like, I don't tune in for the information. I just tune in to watch you guys go. Can you hear me now? Can you hear me now? Can you hear me now? And we've gotten a lot better since then. That's just in general. We've got the savior himself.
Just give me a second.
Oh.
Hey, Peter. How about if I just kill the phone?
Yeah. Yep.
What are you guys trying to do?
We were just having a very small feedback issue. It wasn't having two microphones on. It was just I felt like I was getting a little feedback from his microphone. So we were just trying to work around that.
Yeah, but I think I just killed the mic on the other one, and so we're good.
Yep. Yeah. We're good now. We're good.
Okay. I think. Yeah. Okay.
Yeah.
So I'm in screen share, I think.
Yep. You're back in screen. Okay. You're in full mood. Okay. I put you up. We've got the full presentation on the screen now.
Okay. Cool. Yeah, I can't see anything. If anything comes up, Chad, interrupt me. Feel free.
Yep.
Peter, yeah, all that good stuff. So, quantitative genetics is the analysis of traits whose variation is determined by both the number of genes and environmental factors. This is very important. So, like I was just saying, each one of those individual genes that controls the trait has an interaction with its environment. So that makes it so that there's much more overall environmental influence on quantitative traits as opposed to qualitative traits, which are typically determined by a present-absent sort of breakdown or dichotomy. So, selection. Basic goal is to develop elite genotypes with an outcross population, that'd be cannabis, typically speaking. This is done by increasing the frequency of favorable alleles, which is selecting on additive. This is oftentimes represented by an A in a lot of the different sort of mathematic representations that they do, because a lot of times they'll talk about A plus like additive variance plus interaction would be like additive A plus D, and like there are equations for determining or estimating those effects. In a large population, continual improvement or response to selection is expected over a number of generations. With inbred populations, now this also can apply to cannabis, but this doesn't apply to most of what we're talking about in terms of cannabis. Very few inbred populations exist in the types of cannabis that people are breeding with. So if you were to, however, start with an inbreeding population, you would be looking to generate inbred lines to cross these to develop elite lines for breeding purposes. This is what was done for like heterosis or corn breeding. Here you're selecting on the genotypic values, and this is more of the additive and the interaction. So you've already established that there's an inbred genotype. There's very little variation within the population. So when you're making your cross, you're looking at both the interactions and the additive variance that you're bringing to it. So epistasis is more important when dealing with inbred lines. Further progress depends upon generating new variation through additional crosses. So this is why in a lot of traditional breeding, they've already inbred their lines, or in traditional crops, I mean to say, they've already bred a lot of their lines to inbred lines, and now they're just looking for new variations that they can add or introgress into their population to increase some sort of agronomic or economically valuable trait. We're not really typically at that stage in cannabis. So outcross populations. Improvement in an outcross population this is why animal breeding is a good source of information. Neville was a big proponent of looking at pedigree charts and looking at the different methods that animal breeders have used. Chimera has brought up the point that with cannabis, we're dealing with an obligate outcrossing species. And so when you're dealing with that type of breeding structure, it's much more akin to what's going on for animals. And we talked about a little bit about this last week. So recurrent selection is typically the method that's been used. And in cannabis, we've used recurrent selection for traits like shortened flowering time, increased yield, and THC particularly, but also CBD. Other breeding programs have been more oriented towards CBD or limiting THC, but by and large in the quote unquote drug market or drug cannabis, we've been breeding for high THC, and we'll look at some of those numbers later. So within generation change, the increase in trait mean among the selected individuals. So this has to do with the breeder's equation. We'll get into the specifics, and there's some good charts and diagrams that kind of break this down. But essentially, the selected individuals are the portion of the population that you're choosing to breed with and have reproduce. They're going to ideally have a higher mean than the general population as a whole for whatever the trait is. So if we're constantly selecting for a subset of the population that are high THC individuals and their mean is in the upper percentile, up in the, it was the 10s, the teens, the 20s, the 30s, that process was a process of continually selecting up in that upper gradient and threshold and not selecting just in the overall population. And within generation change refers to that mean difference. This can be translated into generational change, and I just kind of explained that in terms of the gains that have been demonstrated over the decades in THC numbers. Individuals are chosen on the basis of their phenotypic value, so mass selection. This is largely what we've done. Performance of their offspring, so this is progeny testing. If you go and you read Neville's work on, or read Neville's posts about his work on, well, I've posted almost all of them in my stories, but I'd encourage people to go on to the Mr. Nice forum and read all of Neville's old posts. He goes in depth about progeny testing and how you can use pedigree charts and things like this as a way of really doing your selection and improving your traits. Another way of evaluating this is SIBS or family-based selection. So you can grow out a population and grow out multiple different half SIBS or related plants and see the variance within them. And through that, you can do regressions and kind of approximate. This is a lower salience way of determining the heritability and trait performance in a population, but it is a way that is used. And the idea is to use information to obtain estimates for the breeding values of individuals. So this is just explained the breeder's equation, and we'll get into that. So here's the basic model. You've got your phenotypic value, and a lot of the equations it's represented as Z. And So phenotype equals genotypic value plus the environment. So a genotype interacts with its environment for a quantitative trait. There's far much more of this interaction going on because there are more of these genes controlling these phenotypes. This is why, in terms of traits, when doing analysis for and estimating heritability for quantitative traits, you really have to try and control for the amount of alternate variability. So there's variability from the environment. That variability, by and large, the vast majority of it, aside from some poorly understood epigenetic portions, is not inherited. This is what's inherited. And you have to discern this portion of the equation to be able to select and breed and improve your breeder's equation for the particular traits that you're selecting for. So this slide gives us a good demonstration of this idea. You've got variance in your phenotype, which is the result of variance in your environment and variance in your genotype, plus the variance in the genotype environment interactions, which is explained down here, which is variance in genotype environment is a genotype by environment interaction reflecting the fact that genotypes may respond in different ways to changes in environment. So this is why it's important to grow out your varieties in multiple different environments. There's a paper later on that we'll look at, and we've talked a little bit about by Anne-Michelle Faux, and she talks about growing out monoecious cannabis in different environments and scoring the male flower trait expression from, I think, like a 1 to 6. And the point being is that in the different trials that she ran, the expression was different, and that difference was very much a result of this genotype environment interaction that was going on. The goals of quantitative genetics are to partition the total trait variation into the genetic portion, so this is the heritable portion, versus environmental components. Environmental components, largely speaking, are not heritable. Part of it's about predicting resemblance between relatives. We're not going to address a lot of that. That's more of the inbreeding covariance. Sewall-Wright's work is really where you want to look if you're interested in this. This will give you your inbreeding coefficient and theoretical odds of particular breeding programs. The other goals are to find the underlying loci contributing to genetic variation or quantitative trait loci. This is really where we're at now and where a lot of the science is. We'll be looking at a bunch of different papers that have looked at different quantitative trait loci for flowering time, for sex determination, and different secondary metabolites. Deduce the molecular basis for genetic trait variation. That's where a lot of the genetic marker work is going on right now. And then figure out the expression loci. So this right here is a lot of this stuff is looking at the transcriptome and things like that right now, and the different ways that the transcriptome can modulate these traits. This is really just a representation. The main point of this slide is to show here's how 3 genes distribute in a continuous variation. Here's how 4 genes can distribute into continuous variation, and this is how it would plot. here we have a bell curve. This bell curve, I think it's typically called a Gaussian distribution. Each one of these bars represents a standard deviation from the mean. So here's your mean of the population, and each one of these out in each direction represents one single standard deviation from the mean. This is also known as a single Haldane. We talked about JBS Haldane last week. This was one of his ideas, or he helped develop the math for it, so he gets the naming credit. So that's a introduction to quantitative traits. We've gone over, the basic intro there. Now, the breeder's equation, and Chad, feel free to chime in, or any.
Oh yeah, I'm here still.
Cool.
Yep, we're just listening. And one thing, too, on the last slide, you were talking about Loki, and Loki is basically, this is within the DNA. It's in the genome. Do the alleles sit in the Loki, or how does that? I'm just trying to paint pictures for people.
Yes.
When we're talking about this loci, where is that in the plant or in the DNA type? What are we looking at there?
So these two A's would be there, so to speak. And then this would be another loci right here, the two B's. So each loci has two alleles, and that's, the loci have multiple alleles, and
the alleles
represents the different loci that would then be inherited independently.
Okay. And the alleles are going to be the things that are controlling different traits, such as whether you're going to be purple or whether you're going to be CBD dominant or whether you're going to be, that's kind of where that happens, is the allele level?
Yes.
Okay. So the alleles, which are turning on or turning off certain traits. Granted, that's a glossing over of everything. But those are sitting on the loci, and the loci are what we're looking at here. And then the numbers across the bottom are the distribution. You said the top one has 3 different traits, or 3 gene pairs, and the bottom has 4. Is the number along the bottom just the combinations?
Yeah.
Or what does that represent?
Yeah, this is the phenotypic distribution that you get as a result of this genetic pairing. So this right here corresponds to Nielsen L's work with wheat color distribution. So when he crossed wheat, he found that 3 genes were controlling the expression of the color of the kernels of the grain. Mm-hmm. And this was the phenotypic distribution that he saw in the population. And so when you see a phenotype breakdown like this, you're dealing with continuous variation throughout the population that's controlled by 3 genes in this instance. When you've got 4 genes controlling an individual trait, then you would see a distribution more like this in the population. And again, it's continuous, but you're dealing with more genes in a larger subset to get a full representation of the accurate distribution.
Okay. And that's good too. It expands on, last time I kind of learned a little bit more about phenotype. It's more than just genotype expressions. It's really, as on the last slide you said, it's genotype plus the environment. And the environment doesn't necessarily add things or subtract things, but it turns up or turns down that genotype expression to get the phenotypical expression. And then you just added another layer onto that with environmental interactions. How does it handle, okay, all of a sudden it fell to 30 degrees. Well, what is that genotype going to do with that environmental input and express as a phenotype? If I'm hopefully learning as we go along here.
Yeah, no, totally. And like the difference between what will happen for some sort of like Siberian autoflower or Alaskan autoflower, something that's been acclimatized to a cold environment and has adapted to it, to compared to what will happen to something that's just been in the tropics, put in those environments. You know, like that sort of genotype environment interaction is going to be drastically different between those two different genotypes put into
Yep.
those two different environments. And then if you take either one of them out of their, you know, their adapted environment, you're going to see a wide range of interactions that are not just genetic interactions or genetic expressions, but are instead the way that those genes express in the environments.
Cool. Right on. All right. Thanks for clarifying that one for me.
Yeah. Um, So yeah, the breeder's equation. This is really, you know, I like to joke. Are you really a breeder if you don't know the breeder's equation?
Don't ask that.
Yeah, no, that's taboo. But so yeah, in the breeder's equation, this is the most simple representation of it. It gets way more complicated, and it's intense. But simply put, what we're dealing with is response equals heritability times selection. So this represents roughly what's going on here in the breeder's equation. So you've got your population mean. That's represented by this U. Uh, like I said, I didn't take statistics. I don't know the math name for the U. But you've got your mean here in your bell curve population. Then you've got over here, this is the mean on your threshold for your selection. So this is the subset of the population that you're selecting for over here. You're not selecting all of this portion of the population. You're selecting 5 to 10% of the population. There are higher or lower thresholds depending on the trait. Then, in addition to that, it helps to go, well, here, I'll go to this. Really, this is the better slide for showing the full thing. So in the last slide we had, here's your, in this case, random mating population. So here we've got a random mating population. It's not inbred. You've got a continuous distribution of your trait. This is your mean. Your selection threshold is here. In the next generation, what you're going to do is you're going to do the same analysis to try and analyze your phenotypic distribution in the population, and you're going to figure out the mean of this population. The amount at which it's increased for the trait that you're selecting is the response to selection. So, and that is an effect of heritability and the intensity of your selection. So your intensity of selection and the heritability of your trait will determine the rate of the gain that you get from one generation to the next in terms of mean population phenotype expression. Does that all make sense?
Yeah, it's funny. You should see me right now. I'm holding up like fingers. I'm like, okay, so intent selection, meaning I've got a whole bunch out there and I'm going through there and picking it, and then the variability or heritability is the ones that I want. So I'm thinking those. Okay, so things will be better if I do a tight selection only for the things that I'm wanting, because in the future offspring, it's more likely to pass on those traits that I've gone after. Is that
Yeah,
rough?
a little caveat there is that the selection is the amount of the population that you're selecting. So you're saying, okay, I'm going to select this subset. All of these are going to be used in this breeding project.
Okay. So
The
it's
heritability of the trait is the amount of response that you're getting under the intensity of selection. So we'll get in here.
okay.
This is the idea of heritability, and heritability is oftentimes it's a sub, like only a fraction of the phenotypic variability is heritable. And this is actually why phenotype and this whole method is incredibly labor intensive and requires way more work than the new methods, which involve a lot of screening for already identified genetic markers. But essentially, what you're doing here is you're growing out huge populations that have, and you're using a method of selection that isn't very good because you're using the phenotypic expression of this subset of the population. You're essentially saying, okay, let's just arbitrarily say this line right here is the 30% THC. So I'm only selecting things over 30% THC. Everything else in the population is not getting selected for.
Okay. And so
The
yeah,
hair,
you're being
the
super
in,
selective.
yeah, and so the increase that you have in the population in the next generation is the heritability of the trait.
And the increase meaning more 30% or above THC in the next population.
It wouldn't, because of regression, it wouldn't be by choosing these extreme phenotypes, we're not necessarily going to exceed the extreme threshold we've already established. There will most likely be a regression to the mean. And but as long as there's an increase relative to the mean to the previous generation, you've experienced some gain.
Okay, so that makes sense. Like we might not get all 30%, but if the previous generation, the mean was 15, and now all of a sudden in this new generation, after the intense selection of only 30%, if the new mean jumps to 17%, well, that's a 2% increase from the previous generation.
Totally, exactly. That's
Okay,
yeah. And
awesome.
like, if we were to say like this is 10% or like 30% THC, and like maybe this is down 20 or something, or high teens, this distribution here, when you look at this, like you're getting pretty close. You're up in the high 20s for your mean in the next generation. So in theoretically, just looking at this, it would have been, your response to selection would have been very good for the particular trait, which means you have a highly heritable trait. So the concept of heritability is really, there are ways of estimating it. It's a little bit complicated, but it's the fraction of phenotypic variance due to additive genetic value. This is, heritability is additive variance divided by phenotypic variance. That's narrow sense heritability. This is what we're dealing with typically with outcrossing species. Then So phenotypes can be measured directly, breeding values can't. So this is kind of what we're talking about there. In theory, if we had the ability to select the one plant here with the absolute best breeding value and transmit its genetics into the next generation, we would significantly increase this mean every generation, much more so than just using this method of selecting the mean amongst the best. So broad sense heritability, this is what applies more to inbred or inbreeding crops, and goes over narrow sense a second time. But broad sense heritability applies when you're selecting among pure lines. Here you're dealing with genotypic variance and divided by the variance in the phenotype. You know your genotypic values much more on an inbred line, so this is a known quantity much more so than it is over here where you're dealing with open pollination or random mating populations. Here's how you would actually this is how you calculate your heritability for a particular trait. You calculate the average phenotype of two parents, calculate the average phenotype of their offspring, then you graph these points across a set of parents and their offspring. So you're doing a regression analysis and looking for the correlation in between these. This is zero heritability. If your plot ends up looking like this, the trait just simply is not heritable. Here you've got a 0.5 heritability. Even though it's not great, we'll see that's actually a pretty good heritability, and very, very significant gains can be accomplished with a 0.5 heritability. And then this is perfect heritability. It's a 1-to-1, and there is no real, the phenotype and the genotypes match up.
And then the obvious correlation there that I see, just, I mean, looking at the graphs, all the dots on the one with heritability, they're all right on the line. Whereas the one at the top with 0, you know, that's the most spread out of the graphs there. So can that almost be related back to like inbred line would be on the bottom chart, because everything in that falls similar or close to each other, whereas like the polyhybrid cookies would be the top chart because there's so many possibilities as far as what comes out? Or does
Yeah.
is that a fair relation, loose relation?
It is. It's a little bit different just because we're dealing with individual dynamics within those sort. Like that's true of talking about the overall, like the polymorphism or the heterozygosity in the population. Heritability is a little bit different just because it's dealing with the transmission of the particular trait over a generation. And one of the things that's like, you know, you can have a trait that like transmits 100% from one generation to the next, but the heritability is 0 because it's a fixed trait. It's an inbred line. Like you're not getting any gain. You're not changing anything. You're not. It's just, it is what it is. It's stable.
Okay.
But so it's one of those. But it's totally hair. Like it's that's one of the paradoxes, because this is a regression analysis between generations as opposed to an analysis of a individual, so to speak. You know.
Okay. Yeah. Population versus singular.
Yeah.
Okay. And here actually is sorry, earlier we had somebody, let's see, Shredder 0911 was talking about statistics and being able to assign numeric values to each trait that everybody agrees upon, you know, again, kind of setting that standards. And just kind of reference like stem rub, you know, his number may not match what our numbers is, but you do have kind of a few charts in here or something that at least looks to seek to establish some sort of framework for that.
Yeah, we'll get into some of that later. There was a really interesting paper that was recently published. The name of the author escapes me, but they did an analysis of morphological traits throughout vegetative growth, and essentially created a predictive equation for, and obviously a standardized method of evaluating these traits so that you could do this predictive method. But essentially, this right here, I know what it is. It's this paper. The characterization of key physiological traits of medicinal cannabis as a tool for precision breeding by sorry if I butchered their name, et al. In 2021. So this paper is very interesting. They actually were able to determine the heritability of particular traits and obviously they had to do that by standardization. You can't just, I would not end up with these same results if I didn't use the same standardized methods of evaluating the traits that they used. And so that's why it is very important for us to, one, establish what these standards are, and two, normalize them across different research modalities. And to the extent that we who aren't necessarily in like academia or doing these research papers can adopt or develop these methods, or we as a community can at least come to our own shared version of them, yeah, that would be very helpful.
Fair enough.
Yeah. But yeah, I'm not sure if my stem rub numbers are the same.
Yeah.
But so this right here, this gives a demonstration of the heritability of particular traits, some of which are very important, like dry bud weight has a heritability of 0.33, which is actually a relatively high heritability. This right here shows different thresholds. Like in here, it's still heritability is still high enough that you have a high probability of being able to accurately select for the trait that you're selecting for just through traditional methods.
And now, is this like a 0.2 chance out of 100 you're gonna get the same thing? Because that would seem like a really low number to me.
No. So that's
Yeah.
not, yeah, that's not how this is represented. No. So what's going on here is that this right here is, so you've got a 0.33%, like the fractions here are, 1 is the total. So heritability of here is 1. So
Okay.
if this was 1, you would have bud weight 100% heritable, just fucking lunkers everywhere.
Okay.
So having a heritability of 33, that's a third heritable, like that's a good fraction there. That puts you in this range of heritability, which makes it so you can do selection without needing, like if you're down in this range for heritability, well, I should say this range. This is the range where you're doing populations that are fucking enormous. And ideally, you're going to be using molecular genomics to identify the trait and screen the population for the trait so that you don't miss it. Because there are definitely instances with very low frequency and very low heritability traits that if you're not doing, if you're not increasing this accuracy threshold, you're you're not going to get significant heritability and capture the trait in the next generation. So your trait improvement's going to be shit unless you are able to improve your selection accuracy, which is done through the best way. I mean, there are ways of doing it, and we're kind of going through ways of improving selection accuracy later on. But it's the trump card in, I hate that phrase. It's the thing that's changed the game is genomics, because that really improves this portion of the graph. Your ability to accurately select is vastly increased once you have molecular genomics to improve your accuracy. Otherwise, like, your accuracy in terms of selection's great if you're dealing with a highly heritable trait. It's not that hard, but your accuracy goes down significantly. And not like, it's not a linear, but it's like they say here, it's a square root standard deviation.
So wonky. It's not linear.
Yeah. So this is heritability. Heritability for these traits have all been figured out using standardized statistical methods and methods for analyzing phenotypes, and then tracking those over generations. So selection in the breeder's equation is demonstrated here. So selection is this portion of the population that is allowed to reproduce. Selection differential is this difference, your mean population versus the mean of the subset that you allow to reproduce. It's a function of phenotypic variance of the population and the selection intensity of the breeding program. So if you increase your selection threshold, or reduce, excuse me, your selection threshold, you're going to have a much different mean than if you have a much more selective threshold. And then this shows how the whole equation works out. This could be any trait. It can have whatever value. Here you have your trait. It's your mean in the population. Here you have your selection intensity. So consider a population that varies for a given trait with a mean of X1. Suppose some with an extreme phenotype are selected for breeding and they have a mean of XB. The selection differential is computed as XB minus X1. So if X1 equals 5, the population mean is 5, let's just say 5% THC, and XB equals 8, then the selection differential is 3. So this is how you would compute this portion of the breeder's equation to know that you're selecting, the differential in the group that you're selecting is 3 greater than the mean.
And that is going to give you kind of, gosh, I'm learning along here. That's going to give you how far you come with your X and B. The wider the number in between XB, which is the mean of your selection, versus X1, which is the mean of the overall
That's
growth.
what's going to come right here in this next generation. And the XB,
Okay.
with the XB, what you've got is you just said, in my population, this is the best subset, and this is its mean phenotypic value.
Okay.
So I only selected the best, and this is its mean phenotypic value. In the next generation, you're going to select that are only selected from here. So all of this rest of the population is no longer allowed to reproduce and continue going on.
Okay.
Only this group can, leads to this next generation.
And then subsequently, is your goal to get that differential smaller and smaller as you go on through the generations? I'm just trying to figure out where the significance of the differential would fit in, other than it's part of the breeder's equation. But
So
how do you track that?
The key here is that this is just telling you how much better the mean phenotype is for the thing that you like. Here's my population, but I have only selected for this group. Now, the main thing here is that it is assumed that this group is doing what it's doing and expressing its phenotype, its greater phenotypic expression for the trait because of additive variance. So this is a rough approximation for additive variance. That's really all it is. It's saying that this group over here expresses more or better for the trait of interest than the rest of the population, and it becomes a proxy for additive variance, essentially. Does that make sense?
Yes, I think I'm following it, but I know that on further review, it's going to make clearer and clearer sense to me.
Cool. Yeah. So it's really just a way of guesstimating the best breeding value. So we guess that this group has the best breeding value in the population, but we don't know for sure. So we're going to like select a group. Then down here in the next generation, you're comparing the mean of the population of the previous generation to the mean of the subsequent offspring generation that's been selected from these breeding pairs. So here, suppose the offspring from our breeding population has the following distribution with a mean of x2. The response to selection equals x2 minus x1. If x2 equals 6.5, then the response to selection is 1.5 because the mean was 5. So we've got a 1.5% gain in the overall population in the mean expression for the trait. So if we were starting with a population that in this generation had a mean of 5% THC, we then selected the subset of the population that had the highest expression for THC, which was 8%. We then, with a differential of 3, we then looked at the offspring and compared it to the parent generation's mean, and we got 6.5. Thus, we were able to calculate the response to selection and the overall genetic gain from one generation to the next for our trait.
Okay, I see the math there. I see where that adds up now. And because again, the mean from the first generation was 5, which is represented by x1 in the top graphic.
Yes. Yes.
Okay.
Totally. So this is just a regression analysis, and because these right here, even though you're selecting, up at around 8% and only the 8%ers, and even higher if 8% is the cutoff threshold, so it's 8 plus, then in that situation, because of the regression that happens, and because we're just selecting based off of phenotype as opposed to the actual known breeding value and genotypic value of the plant, there's a regression, and we're seeing that regression here in 6.5. So from 8% down to 6.5, even though this is where we were selecting and it was things that were only 8% or greater, we then have, a response or a mean in the next generation that's 6.5, which is a regression back from where we selected from, but an improvement upon where we were in the overall population. So, yeah, one goal of selection is to choose those parents in a sample with the most favorable breeding values. The problem is that we cannot completely predict the breeding value of an individual from its phenotype alone. So we're talking about this. Consider that heritability is the correlation of breeding value with phenotype. It relates how predictive the phenotype is to offspring value. Now, this is the type of equations you'll see. We haven't covered what all of these represent. It's not very important for our purposes. But point is that phenotype alone is not a very good predictor for breeding value. It's all right, and we can get this type of improvement, but it would be much better to get, if we're selecting the 8%er, it would be great to get 8% in the next generation and not have that regression, i.e. pick the best breeding value in the population. So the question posed here is there a better U than Z? Is there a better mean that we can use to look at than phenotype? So there's the mean of progeny. This is progeny testing. We just did that right here. This is progeny testing. So this right here is the mean of Z, and it's helpful and useful information. And progeny testing is better information, but it requires growing out an entire generation to be able to determine the breeding value of the cross you've made. And doing that, it's like in a sizable enough population to get any statistically valid information from it.
It sounds like the stuff like Breeder Steve is doing down in Uruguay or wherever.
Exactly. Yeah.
Okay.
And last week we had a pedigree chart from Robert Clark and David Watson talking about breeding skunk one. And it goes into, it at least lays out the process that you would be doing here with this type of progeny testing. You can also do weighted mean values of other relatives. We talked about this earlier. You could do like half sibs or siblings and grow out all the different populations, and then weight the values that you get from those to determine or to help inform your calculations on the overall means that you would use to calculate the heritability of traits. And then this one, mean values of relatives grown in a variety of environments. So this is also another key factor. Multiple environment trials help inform your overall heritability or your understanding of heritability, because if you're able to grow out, just as a for instance, this is individual plants, not populations, mind you, but you know, every hype clone has been grown out in 1,000 different environments at this point, and there are lots and lots of different COAs that kind of represent a range of the potential expression of these different phenotypes in these different environments, or genotypes in different environments, and the phenotypic expression that is demonstrated or exhibited by these different clone onlys. So you could take Cushman's and look at the range of variables that it has in different environments and determine the amount of environmental variables or influence that are impacting that particular variety that are then used in breeding. So this will help you reduce the overall noise in your evaluation or in your equations, because you're controlling for some of that environmental variance. If you're able to say, okay, Cushman's produces between like 27 to like 35% THC or some shit I've seen. And granted, I understand there's all the issues with the test. Let's just pretend this is accurate. And
Yeah, we'll just go with the numbers that they tell us for now.
Exactly. So if you have a range of 27% to 35%, and it's an individual cultivar, there's no genetic variance there. Granted, I understand there's viral issues and viral load and those different things. And there was one paper that I'm trying to search for in my archives that is very interesting on the subject of mutations within clonal lines. But point being, you don't have any genetic variance there. You only have environmental variance, and you can then determine the mean there, and that mean at least lets you understand that across multiple different environments, here's my mean for this particular cultivar that I'm breeding with. And you can do that for a lot of different cultivars that already exist. And the more that you have like networks of people who are sharing and growing and working on different cultivars and doing it in sort of a community way, you can really start to see the range of expression of particular cultivars that are being passed around. And that can help you account for the environmental variables that are going to impact or influence the phenotypic expression. Because again, phenotypes are cool, but they're not genotypes. They're the result of a lot of environmental variables and a lot of interactions between genes and environments. And just looking at that phenotype alone isn't sufficient to actually determine its breeding value.
And that does put a lot of time and effort into true breeding or proper breeding practices to, because you can't speed this plant up. and you have to try them. You mentioned COAs, certification of authenticity, which, again, we'll go back to Joe Bob in Minnesota with the official Sour D, the originator guy. All hypothetical again, guys. But yeah, if he's turning out, COAs on his particular cut, he does need to know how it handles the humidity in Florida and the dryness in Colorado and the moisture out here in Washington, because you would get different expressions. And if you don't know it, I mean, not to throw a split in the tracks here, but that's a lot of the talk about OGs and Kush is just kind of like different variations in different areas of the country.
Exactly. Exactly. And like that's OG is a great one because it's one of those, it's a cultivar that people all across California have been growing for, decades. And so we all have our own different experiences with it. And, it's great to hear people talk about OG who are like really been growing it in their area and are just adamant that it's different, and like not necessarily that it's a different cut, but just like everybody loves their herb and, growing it and talking about the expression of the traits. And it's one where when people talk about the environmental variables that produce particular expressions of a cultivar that we've all grown for so long, it's really enjoyable to hear. And it's something that, like, I'm only talking about the different microclimates from the Emerald Triangle to the Sierra Foothills to the Central Coast down south. Like every one of these different places is, got its own little microclimates, and we've all rocked the OG really hard. And there's some people who swear it just loves that coastal environment. Other people who are like, no, it needs that really dry, arid, some cuts perform better, like, the Fire OG versus the Tahoe, etc. Like it's definitely environmental variables are really unique for us to look at just because we operate off of a population that is so much based. There's very limited genetic variety or variation going on when we're just passing cuts around. So we get to see some of these environmental influences that you may not see in other plant species as much.
Cool. Thank you for elaborating on that one, because that is one that I do, again, here as environmental conditions coming into the conversation when you're talking origin stories.
Yeah. Okay, approaches to estimating heritability. So we've gone over a bunch of these, and we've gone over the parent-offspring regression and addressed measuring a population response to selection across generations. It's similar business. We did not talk about covariance among relatives. Mostly it's the inbreeding coefficient. It's Seawall Wright's work from 1921. Check it out. It's very interesting theoretical work. So this is one of the reasons why we didn't actually get into it, partially. So actual relatedness versus expected values from pedigrees. So values for the coefficient of co-ancestry and the coefficient of fraternity obtained from pedigrees are expected values. These are theoretical. They're not actual. Due to random segregation of genes from parents, the actual value or realization can be different. So random segregation, and another factor here is linkage. There's issues of linkage means that you don't have just like a purely, not everything's moving around according to the theoretical principles that Seawall Wright laid out in his 1921 paper. So this gives theoretical, like an example. So you would expect that a full SIB to have a 0.5 inbreeding coefficient, or of the full full subs. Yeah. However, one pair of the SIBs may actually be more similar, 0.6, and another less, say 0.3. So that 0.5 of full SIB population in their inbreeding coefficient, it's the middle point between these two, so to speak. It's an average. On average, okay, sorry. The best way of talking about this or thinking about this is the backcross example. So you cube it, you backcross it 4 times, it's 99% the parent. This is where this type of relatedness value it falls apart, and it's because of linkage and linkage drag and the connections of traits on or of the different alleles on the chromosomes. So the way that you overcome the fact that this is just a theoretical way of determining relatedness or covariance, and the way that you overcome some of the limitations and the labor intensiveness that we've been talking about in terms of having to do the parent offspring regression and looking at the response to selection over multiple generations, which requires growing out hundreds of plants over multiple generations and phenotyping all of them and doing mean analysis and regression analysis for all of these and correlation between the parents and the offspring. That's a lot of work. That's time intensive. And it's also, as we already looked, it's not perfect. It doesn't tell you the breeding value of the particular phenotype that you're selecting, or the number, if you're selecting everything over X percentage of THC, the mean of that grouping, that mean doesn't tell you the breeding value. So genomic selection uses this extra information, and that's really where we're going to transition to with the rest of the discussion. So yeah, we went through a lot of shit. I'm surprised that we got done with all the mathy stuff.
Yeah, definitely got through the mathy stuff. I'm still here. I'm still taking notes, and we're making sense. So, and I know chat is following along too. Somebody had brought up, it's like algebra all over again. It totally is like algebra, but I'm more interested in it this time. So that makes a big difference.
Yeah. Had they only brought the pot plants out.
I know. Well, I talked with Dr. MJ Coco Wednesday. He happens to be a professor as well, so he gets to teach people, and it's amazing how you can motivate students sometimes.
Yeah. No, totally.
But yeah, so back to you. You okay there? Don't choke live on camera, please. I'd feel real bad.
Yeah, no, just puff and tough while we go through. So yeah, these are some traits and some authors who have written different papers on the quantitative trait inheritance and expression of sex determinism, cannabinoid ratio and quantity, terpene ratio and quantity, days to maturity and flowering. And I think I left these ones out because they're hemp related, and I don't really
care. But they're cousins.
Yeah, check it out. This Campbell is different than this Campbell, I believe. And this is Brian Campbell. Brian Campbell worked for Phylos. I don't know if he still does, but this is information that he did in his dissertation, and there's some cool stuff in it, even though it's still fuck Phylos. But he definitely did. He did good work in his dissertation, and has published a good paper as well.
Yeah. Yeah. It's not. It wasn't all a loss. Wasn't all
Exactly.
loss. And actually, we did get one question right here since you're lighting up. They can't actually see it right now, but Culture Rebel had asked what you token on this evening.
It's been a number of things. Uh, there was a really heavy Afghan leaner on the PKM10. Um, then there was a gas leaner on the PKM10. Definitely preferred the gas leaner. And then I did something that's kind of a mystery. I don't know what it is. It's got some weird jack terps that are fancy jack, as I like to call them. It's like terpenoline plus something that's got like, I don't know, it's got like tropical fruit flavors that I really like. I'm pretty sure
Okay.
it's a Wilson cross. But yeah, it was, I liked it. And now, yeah, I don't know what this one is.
All right.
I rolled up like 5 joints before we started.
Always
Because I knew we
prepare.
were going to hunker down.
Yep. All right. Cool. I'll let you get back to it there, but hopefully that answers that for you, Culture Rebel. I'm what am I on? I'm about to reload. I think I'm going Temple Runs. But all right. Enough about me.
Nice.
Yeah.
Okay, so sex determination. There are some really cool papers, and I'm going to talk about some of them, cite them. We can get down and talk on, like, really dive into some of this stuff. But I'll also try and get it all together in another, like, folder link thing, and people can hit me up for it. So, sex determination. In 2014, Anne Michelle Foucault, she published a series of papers as part of her dissertation examining sexual trait expression in monoecious hemp. Um, Key things that she found were she confirmed that monoecious hemp plants have XX chromosomes. So if you have a hemp plant that produces both male and female parts, they cytologically, the chromosomes, are XX. They don't show up with a Y chromosome. So essentially, you have male flower expression on a plant that is chromosomally completely female. It's only got an X and an X chromosome. Faux also identified an autosomal QTL. QTL is a quantitative trait loci. This is all of the discussion now is focused on these things. So this is quantitative trait loci are the many different loci where there are alleles controlling a particular trait. They're distributed across the chromosomes, and all of these different genes at these different loci influence the expression of the trait. And together, they're a QTL. So she identified an autosomal. So you have your sex chromosomes, and then you have the other chromosomes. All of the other chromosomes are the autosome. So in cannabis, it's like diploid 2N X 10. So I think they're like 10 chromosomes, but it's a diploid plant, so there's like it's really 20. Anyway, point being is that I lost my train of thought. Yeah, chromosomes are goofy as fuck.
That's a good way to pull it back. Yeah, you're getting into diploids and the combinations of 10, and then now you've got 20, then all of a
Well,
sudden it
that's what fucking
compounds.
it's the numbers. Yeah,
Yeah.
yeah. So we will get into that, and that's not all that important. The point is that what she found was that in the autosome, i.e. the parts that are not the sex-linked chromosomes, but are the rest of the genome, in those areas, she found a QTL that is associated with male flower expression in monoecious hemp plants. So this is really something that's associated with herming. And it's also tightly linked with genes associated with photosensitivity, which is an environmental factor that we all know contributes to herming. Light leaks, things like this, disrupting the photoperiod. These are things that can cause the plant to start throwing out some male flowers on an otherwise female plant. So her work helped to kind of first establish this QTL, first establish that it was a QTL and that it existed on the autosome. This was a really important paper. It didn't have a lot of, didn't have the most dense resolution in terms of genomic data. So there was questions about it after it first got published for a while. But it got confirmed in 2020 by a paper published by Pettit et al. And they confirmed and expanded on Fo's findings, confirming both that sex expression is controlled in part by QTLs and that these QTLs are linked by genes associated with photosensitivity. They specifically found, I think they looked at six, we'll look at it, but they found some very interesting QTLs and really confirmed what she had found. Then this mtDNA research looked at the evolution of diose in cannabis and reveals a very complex evolutionary history. The most simple hypothesis would be that CMS, or cytoplasmic male sterility, would be the explanation for how there was divergence in the sexes and how diose evolved. However, doing the mtDNA research, they weren't able to. They can't completely rule it out. There's a possibility that CMS still explains the evolution of male-female divergence in cannabis, but it looks like it's a much more complicated story. And the much more complicated story seems to correspond to what we all know with the plant. The plant has a number of different environmental variables that can cause it to start throwing out male flowers on otherwise female plants, and it's probably a result of this complex adaptive evolution of diose in the plant. It wasn't just something that was a more simple and straightforward process, but was instead one that was a lot more complicated. And also, if they don't find the ability to, like, if cytoplasmic male sterility just isn't in the genome at all and doesn't operate in cannabis, then they won't be able to use that the way that they have in corn breeding, which is part of how they controlled breeding, or it essentially facilitated the breeding process because they were able to no longer have to do very labor-intensive breeding experiments by using CMS. In cannabis, it has pretty obvious applications as well for commercial application. Then this paper, 2020, et al. Investigated the transcriptome data to identify genes associated with the expression of male flower in cytologically female XX plants. They identified multiple candidate genes, I think 15 overall. So here's the shit. I should have been doing this the whole time. This is the faux paper. This is the key findings that they had, or that they made. Sex expression in monoecious hemp varieties are quantitatively, very quantitatively. So essentially, through this work and then through Pettit's work, we're now at least we've identified different genes that are associated with the susceptibility or the propensity for female, otherwise chromosomally female plants to produce male flowers. By being able to identify these, we can select against them. We might also find that selecting against them has unintended consequences. And part of that has to do with the fact that it's tightly linked to perception and transduction of light and transcription factors well known to regulate flowering, which were identified in QTLs for flowering time traits. These genes are involved in the regulation of different phytohormones, specifically auxin and gibberellic acid, were identified in the QTLs for sex determination. Sex determination QTLs were associated with the development of male flowers in female and thus female plants, and thus with sex stability determination in monoecious plants. So this paper right here basically outlines the fact that there's links between light sensitivity and the mechanisms that control sex determination in female plants. And so, again, we don't really know the degree to which we will be able to break those linkages because those linkages exist. And furthermore, we don't know the consequences of breaking those linkages necessarily. There may be unintended consequences to selecting against terms on that level, which is one of those paradoxes.
Right. And I could definitely see that, with a lot of just even like typical medicine for, humans, like uptake inhibitors, things, well, we're blocking something in our brain. And then it's years later we find out how the system really functions and worked together. So yeah, you can stop this particular chemical from flowing in the brain, but little did you know it's responsible for some other necessary function of the plant or of the human in this case. But that Pettit et al. Study, that would be interesting. I've got that one written down here. So I mean, they have identified the QTLs and the photosensitivity, because that's been one question I get a lot, and I don't expect you to have the answer here, but in the paper I'm going to see if they touch on it. But a lot of us, you could say it's bro science, whatever, but photosensitivity, light leaks, we know that could cause herms. But then the other side will always come back. It's like, well, bro, I grow under a street light, or like there's outdoor plants that grow under a full moon. And so the last I heard was there was a study that was looking into the intensity and the spectrum and duration of light to trigger that type of effect. But essentially what they would be looking at are these QTLs is what it sounds like.
And yeah, and they're looking at that gene environment interaction, because there's all these different genes that are interacting with those environmental variables, and all those environmental variables are also variable, like the spectrum, the intensity, duration, etc. And so all of those variables then interact with the different multiple variables that exist on the genetic level, and these are like, there's multiple genes, and these genes are all linked together. And so that's why it's hypothesized by both Pettit and Faux that this is an explanation for why you see these environmental reactions or these responses to the environment in terms of harms. And also the Faux paper is very interesting, and I believe Pettit, they both look at these variables over multiple environments. So you do get to, like, if you really dive into Faux's dissertation, you get to see how all the different stuff that we talked about in the first half is done by a scientist to look at a particular trait, evaluate that trait in relation to several other traits, because she was working in hemp. So I believe the other traits she was looking at were flowering time and yield in fiber. But her big, the breakthrough, or the headline findings that she made were much more based on sex determinism. This is the mtDNA paper looking at trying to figure out how diosy evolved in cannabis, essentially. And yeah, as I had mentioned, their results revealed no clear evidence that different reproductive patterns are due to easily identifiable CMS mutations, cytoplasmic male sterility. Our results refute the simplest hypothesis that there was a single recent origin of diosy in monoecious ancestor. Instead, the story of evolution of diosy is likely much more complex. Furthermore, explanation of the nuclear and mitochondrial genomes and their interaction is required to fully understand the mating strategies of cannabis. So there's still a possibility that cytoplasmic male sterility genes could be found in cannabis and could explain how females are female, essentially, and don't produce any male flowers, or, as relatively few male flowers. If those genes are identified, that will be something that is exploited. When we talk about GMO, if no cytoplasmic male sterility genes are found in the interaction between the nuclear and mitochondrial genomes, the end result is that somebody's probably going to put it in there. And so this right here is a target for potential genetic modification in the future. Just throwing it out there.
Interesting. Yeah, bookmark that one. We'll see when that happens.
So the Adal paper is really cool, because this paper actually looked like I think they used STS or some sort of chemicals, but they basically induced male flowers on female plants and then did the testing of the genomics on the actual reversed male flowers to try and see what the genes that were expressing were responsible for the expression of the male flowers. So they revealed over 10,500 differentially expressed genes, of which 200 potentially controlled masculization in female cannabis plants. These genes include transcription factors and other genes involved in male organ, i.e. anther and pollen development, as well as genes involved in phytohormone signaling and male-biased phenotypes. The expression of 15 of these genes were further validated by qPCR assay, confirming similar expression patterns to that of the RNA sequence data. These genes would be useful for understanding predisposition to harming, basically. So this paper is very, granted, it's limited to, I believe, an individual cultivar. It's not like looking at all populations. Or a large population, and it's using chemicals to induce male flower expression. But by doing that, you're able to see analyze the transcriptome, the RNA sequencing data, and understand what is essentially how are these genes expressing in a way that's producing this male flower in this female plant. And they found 15 target genes. So that gives us 15 targets to either, eventually someday select for or against, Because the crazy thing in hemp also, the reason why Fo was doing her analysis of monoecious, sexual expression in monoecious hemp, was because the point was to make herms.
Huh, that's interesting. So to counteract the potential of herms, these 15 genes are definitely of interest. Would these 15 genes also be of interest for people trying to build sterile plants, like sterile females?
Um, Maybe, not really,
Okay.
not necessarily, because these genes are more focused on the ones that cause the female to produce the male expression.
Okay.
So these are the ones that are, these transcripts in particular are the ones that tell the DNA, don't don't go female and produce, all of the different trait architecture and plant architecture for a female flower. Go in the direction of producing a male flower.
Okay.
And
Okay.
so these genes are ones that are much more likely to do that. And I mean, the fact that they were able to find 200 genes that potentially, not all of them, but potentially control masculinization of female cannabis plants, like, if we're dealing with 15 genes, that's a fuck ton of genes. And if you look at the number of, like, when you start pulling out your Robert Allard or your Seawall Wright and analyzing what your selection intensities have to be when you're selecting for or against 15 genes, I mean, it's fucking crazy. And 200's like bonkers. So, we're dealing with pretty large quantitative traits here. There's a lot of genes that play into both the morphology aspects, but this shit in particular, the phytohormone signaling, all of the male bias phenotypic type expression. We were talking a lot about this stuff on the breeding roundtable this week, and specifically how some of these male bias phenotype or trait expression is things like stretch, and like why gibberellic acid is used is to try and induce that expression or that extension in the plant so that you actually get what, male flowers are much more extended. They're not really tightly compact nodules. And so part of that is just trying to, chemically manipulate the plant using exogenous hormone to induce a response in the plant. But these genes, once you start getting into signaling for hormones, you start involving a lot of different metabolic pathways and different genes that are going to be involved. And that's why these things are just, it's so much more complicated than talking about Mendel or, most of the breeding books that we look at, because once we start getting at this level, it's either a lot of statistics or PhDs and people with lab coats who you pay money to basically run these qPCR assays.
Right, right. Okay, okay. That makes sense there.
But it's still, I mean, it's knowledge, and we can apply this stuff still. And this gives us an idea, like the fact that we're honing in on on the genetics that produce male trait expression in female plants gives us way more saliency just on how this shit works, because that's so much, like, that's incredibly valuable, and it gives us the ability to understand that if you have herms in your population, that doesn't mean that you can't use these methods that we've gone over to select against that, to move away from those traits and to experience the gains. Like if we're talking about a mean expression of 5%, let's say, of your of the population is expressing herms, and then you select away from that constantly, and you're able to look at those mean regressions. Hopefully you're able to, and talking to people who have done long-term breeding programs to try and get away from herms and things like GG4, Gorilla Glue 4, or different things like that, they've done exactly that. They've intensively done negative selection against herm traits and for non-hermit expression and experience significant gains and moved away from hermit. Or David Watson talks about it for the Durban poison and breeding that away from a very intersex population to a much more stable population.
That's interesting. I didn't know about that, but Durban poison's rumored to be one in the lineage of a few strains that I kind of, I don't know. I make fun of cookies a lot, and you know, there's many reasons for it, but I like to knock the herm factor of it. And I actually see Flora Nugs in chat right now. He's a person who sends me lovely shots of cookie strains all the time. Like he's got the good ones. But to bring it back to the point, you know, I had finally, after years of asking different people the same answer, James Loud on his show kind of gave me the answer of like, well, you know, it can be bred out. You know, exactly like you're just saying here. But for me, that was kind of the first time I heard it because, you know, we'll edit this out at the end. Not that we can really edit, but you know, cookies have some really fire and stable stuff. And it wasn't always that way, and there's still a lot of it that sucks. But no, there are some that are totally stable, whereas before it was like, you know, a new strain every run because you got fresh seeds. But yeah, so knowing this is the way to statistically or use data to breed away from those things, which is exactly what he was saying probably happened with a few of the better breeders working that line. Okay.
Yeah. And also, I should point out that they found 15 target genes here that were associated with male flowers and female plants. That doesn't mean that every one of these 15 genes is necessarily the most responsible or the best target for addressing or breeding away. Some of these, you're going to have a much higher heritability in terms of your ability to breed away from them than others. For instance, if the heritability on this subset is different than the heritability on this subset, your selection here is going to be much more effective than your selection here. And you can go for this trait. They talk about this in cannabinoid inheritance, that it's possible that copy number variants, the number of THC synthase genes that are in the overall plant, that those could explain the percentage of THC. It doesn't necessarily explain it, and that one of the better explanations, instead of the number of variants that synthesize THC, you need a lot of the precursor, the things in the upstream in the in the metabolic pathway that make it so that the plant can produce THC in the first place, because THC is the end of the metabolic system, so to speak. You know, that's one metabolic pathway, and it's kind of the conclusion. It can go THC or CBD, and those are the end results of a lot of different metabolic exchanges that happen earlier. And upregulating for those other genes may be more effective for increasing overall THC. And same thing here.
Yeah, okay. And it does. It sounds like, you know, again, I don't understand all of this, but if it follows a similar pattern, you know, those 15 genes, it could be a Rubik's Cube of combinations that bring out the expression. Kind of like you're saying, I think you're saying it may not just be one particular one, but there always seems to be variables with all of these. So it's probably, again, a Rubik's Cube of combinations there.
Oh, yeah. Okay, so that was mostly male trait expression in otherwise female plants is a quantitative trait that exists on the autosome, and, you know, ideally people are selecting against it, but it's incredibly complicated, and there's some serious linkages there to other traits that are key to, you know, the overall function of the plant, like flowering time and sensitivity to light. So terpenes. Terpenes are, like we said, one of those genes or subset of genes that exist in a whole lot of different variety or species of plants. Broadly speaking, there are 6 subfamilies of what are known as TPS. Terpene synthase genes are abbreviated as TPS. These TPSs are the subfamilies are listed A through F, so it's TPS A, B, C, D, E, F, G, or excuse me, F. There are over 35 TPS genes that have been identified in cannabis. This is a really interesting fact. Individual TPS genes can produce more than one terpene expression. Oftentimes they produce a major product and then minor products, so it'll be it primarily produces myrcene or pinene or terpinolene, but then other terpenes will be minor products that are also created by these genes. So this is like a much more leaky THC synthase sort of idea, where in THC CBD ratios, it's understood that even when you have, quote unquote, pure CBD varieties that only have non-effective THC gene synthase, for whatever reason, there's still minute amounts of THC that end up in the products or the end field results, and those can sometimes trigger, hot batches in hemp. That's from the leaky synthase gene. That's going on in terpenes as well. So terpenes and cannabinoids rely on similar metabolic pathways and precursors as well. So there's potentially, I believe, Chimera and NAPRO did some analysis, and they saw like a 15% variance or correlation in the variance associated between these two. So essentially, the cannabinoids and the terpenes are competing on some level that can express, or is represented by about 15% of the variance, but then the other 85% of the variance has nothing to do with the fact that these are essentially competing over the same pathways and molecules. And space as well. So then linkage maps elucidate genetic architecture of the TPS genes, their location on chromosomes. So we'll see some of these at the end. So here's a bunch of the literature. Terpene expression and inheritance is part of a complex set of traits that are quantitatively and qualitatively expressed and transmitted across generations. Chimera was talking about this. You will still see discrete segregation in terpene values, so they do kind of differentiate, similar to your quote-unquote traditional Mendelian dynamics. And if you there are some terpenes that aren't found in certain varieties because they haven't been bred into them. Theoretically, they could be bred into them, but the non-existence of a terpene can make it so it's a discrete segregating population that doesn't necessarily conform to a true pure bell curve. But there are like I mentioned, there are over 35 terpene synthase genes that have been identified in cannabis. This is the literature. We'll go over a bunch of it. This is a nice graph that was made by NAPRO and published in their 2016. It was a slideshow that they somehow I found it. I don't know where I found it, but anyway, this shows maximum terpenes was 2.4%, minimum was 0.6%, average 1.5. Okay, so shows different variations from the various lots. Terpene variability is greater than cannabinoid variability. Here's the Booth paper. Booth, Ryan mentioned Judy Booth. Her work from 2017 to 2020 has really been, I think she's done the majority of terpene synthase genomic work. She's identified, I believe, 35 different terpenes or TPSs. So this was her early paper in 17. She looked at phenola, which is a hemp variety, and found 9 cannabis terpene synthase genes in just 2 subfamilies, A and B. So yeah, and these are the specific myrcene, ocimene, limonene, pinene, caryophyllene, and humulene. She expanded on this work and did an additional analysis on Purple Kush. Purple Kush, the genome was published by Van Backel in 2011, and it's been the subject of a lot of analysis because that genome essentially has been available to people to then do the, like we said, these terpene genes. They're a family of genes that exist in a lot of different plants, so you can look for the analogs in cannabis, and she's done that, and she was able to identify 33 different cannabis terpene synthase genes in the Purple Kush genome. So yeah, this paper, this did a lot more work and really developed the overall number of genes that had been identified. Then her dissertation is really worth reading. I would check this out if you, well, I mean, I've got links for it. It's freely available. But here she goes very in depth and looks at a lot of different terpenes, and yeah, I think it's 6 chapters. So, and it goes further in depth and talks about more varieties and more terpenes. And I believe there's 1 chapter on cannabinoids. So Booth's work was like foundational to terpene synthase genes. This is a way out of focus picture, but this shows the idea that different specific terpene synthase genes, TPSs, have both major and minor products that they produce. So the major product of cannabis, CS is cannabis, TPS is terpene synthase, F1, FN is a reference to phenola. So the major product of that gene was limonene. Minor products were pinene and alpha pinene and beta pinene, as well as myrcene and terpinolene. She goes on to demonstrate this with a number of different genes. And as you can see, each one of these genes not only regulates or produces limonene or these other terpenes, or excuse me, it also produces these others. So when you have, you're not just inheriting a limonene gene. When you inherit this gene, you're inheriting the production of as many as 4 to 5 other terpenes as well that will happen in minor amounts. So, and then, you know, this is where you really get into a very, very complicated inheritance scheme and a very complex additive. This is where you have to do variance analysis, and that's where you're going to need to look into R.A. Fisher's work. And analysis of variance is an idea that he developed, but there's a specific set of mathematical principles that actually help you break down the fractions so that you can calculate for the major product that's produced by these different genes, as well as their different constituents, as well as the fact that their different constituents are all different. You know, here you've got a major producer of alpha pinene with minor amounts of these, and then you've got this one producing a single detectable product. So this one only produced myrcene when exposed to GPP. GPP is a precursor that's involved in the synthesis path, or the metabolic pathway that's synthesizes terpenes. So anyway, this really shows, even though it's kind of blurry, the incredible complexity of the trait expression of terpenes.
It would seem so, because there isn't a consistent factor there. You know, at first with the TPS2, you know, it made a major and then some minors, and in my head I'm kind of like, okay, it's more of a factory than a mold. You know, it can put out a bunch of things versus one specific, but then the TPS3 specifically to myrcene, so then that one would be kind of like the mold. So yeah, just does go to share or show a lot of the variability in these types of things. And if we get to a good point, have one a good question in chat about the masculinizing genes. Masculine. Good chance to interject that one real quick. Okay, awesome. And you know, it just raised my head. It was Micah Perry said if the community bred, and you know, if we go in and we do you know, take a look, and again, we're assuming that these 15 genes are the, or well, 200 genes actually. There were 200 genes in that study for the masculinizing. Okay, I'll give up on saying that word. But the question was, if we as a community bred out the masculinizing genes, would that mean that reversing females for pollen wouldn't be possible anymore? Or at least would it make it more difficult?
Yeah, it would.
Okay, perfect.
Yeah. Because I mean, these are the genes that they found doing exactly that process. So if we selected against those genes and removed them from the population, we would potentially be eliminating the possibility to do reversals. Now, you know, it's a very, this right here is a wild beast. You know, once you start dealing with phytohormones, you know, plants are, I mean, fuck, I haven't looked into it, but I just saw that condors asexually reproduced. I don't know if it's true, but if that's fucking true, it's fucking crazy, because supposedly there was males in the, like, mating population nearby, so, like, theoretically they at least could have mated, but they chose, or, like, you know, figured out somehow to asexually reproduce, which is the first time in birds that's ever fucking happened. Crazy.
Yep. And if you've ever watched Jurassic Park, they will find a way. But that's interesting though, because again, I know they're looking at breeding plants with no, you know, that can't get pollinated. And then if they were to remove those particular genes, then somebody truly could put something to market that you couldn't reproduce or copy. You had to buy fresh seeds every time.
Yeah.
Okay. In my grand conspiracy mindset there. Okay.
Yeah. So that's, I mean, that's a really, really great question and point to bring up. That, like, theoretically, if you were to select particularly against that subset of genes that made it possible for that expression to happen, yeah, you would be eliminating the ability to reverse using, I believe it was, I'm off the top of my head, not having the full paper in front of me, I'm pretty sure it was silver that they, silver thiosulfate that they used for the reversal to then do the experiment and see the transcriptome genes that were associated with it. So Livingston basically builds on the same thing with Booth, really looking at different specific terpene synthase genes, ones that produce osamine. All of these are very similar, honestly. Zanger in 2017 has also was able to identify and evaluate some other genes that hadn't been identified, specifically this the linalool and nerolidol. Yeah, and these ones down here at the bottom, these are the genes that they specifically identified in their study that were different from the genes that had been identified by Booth. And so with all of these different genes taken together, you've got I believe we've got a composite of 30 different target terpene genes that have been identified. And theoretically, because they've been identified, markers and assays could be created for these. So essentially, you could identify the presence of these genes in a vegetative plant without having to grow it all the way out through completion. Granted, as we've said, environment is a massive variable in the expression of these traits.
Mm-hmm.
So, like, take that for what it's worth. But at least knowing the presence or absence of the genes is possible.
And that is interesting on the surface, just for the simple fact that you know, or you could get a good picture of what the profile is going to be without having to grow it out. And if that's not something that is hot on the market or whatever, then toss it. Or if it's common, toss it. You might be able to find the outliers that way, and you keep it and you keep running it, because again, it's people with budgets that are going to have information or this type of information at hand to make these type of decisions. It's not me in my closet.
True, but also as I was sitting here thinking about it, I was like, Steep Hill back in the day, a decade ago, they were fucking using the Mandolino and Shoyama fucking papers on markers for male sex expression. The male, I think they're called MAD or MADC. Basically, they found markers associated with male sex expression, and that's what the first sex kits were based off of. Essentially, fucking Steep Hill, yeah, they had some money, but they didn't have crazy money back in the day. Like 2008, 2010, whatever it was, like they weren't fucking. We're not talking millions and millions of dollars in startup funds. Like they had some rudimentary lab equipment, and they went and they did it, and they did it using studies like this. So if you were able to acquire the different lab equipment that is needed to run the PCRs or qPCRs, you could do these assays in-house, and you could screen your own populations and find these genes. Or you could also make a fucking, like, a company and sell your, like, so much. I wanted to, like, mention this, like, so much of the gene editing, biotechnology, patents, 90% of that's just a fucking shell game or like a pump and dump strategy where it's like, look, we've got value. If you build out a company and you have the lab equipment and you build out the company and you provide early terpene testing, perhaps Medicinal Genomics is doing it. I'm not entirely sure. There's a number of different people in the cannabis space, quote unquote, that are offering sex kits, etc.
Mm-hmm.
They're right now, they could take the same information and start developing these marker assays and doing this for people and being able to tell you whether or not you've got the synthase for linalool. That's not going to tell you yes or no you've got linalool. That's just going to tell you whether or not you've got this specific terpene synthase.
Well, that's cool. That sounds like it's a lot closer for us as a real possibility then to benefit.
Totally. Yeah, and I think, check out Medicinal Genomics, because they had qPCR kits where, like, instead of sending your sex kit into Phylos or some other company, they would essentially send you like a test tube kit, and you could do this like little lab experiment at home and determine the sex of your plants. And there's ways, and like my homie Bam, Bollywood Bam, he was somebody who I did a lot of this early studying with, and has actually done the lab side of things. I didn't do the lab side of the courses, but he has, and he has experience with that stuff, and he treats it like, we talk about this stuff, and I've for a long time been like, oh man, that's an extra next level shit, and he's like, nah, dude, like, just fucking get your lab fucking protocols and your fucking cleanliness on point. Don't fucking contaminate your shit, and you'll be fine.
Huh. Well, yeah, and that's I think kind of where Steep Hill came out running. Without the super millions of dollars, is they had passionate people who were really really curious, and they didn't have some answers to things, and they kind of blazed their trail. I don't know the full story, to be completely honest, but just from an outsider perspective, that's kind of, it wasn't the shell game. It wasn't like, okay, if we get this building, we get all this equipment, then we can sell it to an investor because we got the license. They were actually trying to solve something.
Yeah, no, it was like, fuck, we have to take investors. Fuck, this really sucks. Investors are the worst. Why can't we just do cool shit?
Yeah,
And
yeah.
I'm a scientist, like, because Reggie, and I forget the older dude's name, but like, they had cool cats there that like you'd go in and you'd talk to them, and you wouldn't just like talk like, now I'd be stoked to go in and talk like sciencey shit, but like you could talk weed shit with them, and they were like heads, like they knew just as much on that side of things as they did on the science side of things. So it was like early Steep Hill days were really cool in terms of the scientific team that was there, and like a little over a decade ago. And Reggie was one of those people, and Bam, he did work there in terms of just like doing some lab experiments, and the Holy Banana work that really actually the the freak show came out of a breeding population that Bam did that was run through some genetic testing through Steep Hill, and it was part of an experiment that he had constructed to try and identify the cannabinoid, the THC synthase of the particular population they were working with, which was Holy Banana.
Wow, that's interesting, because that's something that I have in my tent right now, is a couple freak shows, and while it was slow to grow, it was just fun. It's like a Guar concert. Everybody should do it at least once. Everybody should do it once.
Twice.
Okay, there you go. I go twice.
Yeah, no, Guar, greatest. Yeah, it's top 5 easy greatest shows. Yeah, but so no, and we're hopefully working on bringing on Canna Research and going to talk about Freak Show and stuff like that, because that's all part of that time period, and that those plants ended up at the farmers market. Like basically, Bam did a huge population. He had leftovers. He wasn't going to grow out all of them, but had done like some of the genetic sampling from them, and then like hit people up on the gram and was like, I got all these, like, get at me. I'm going to be at the farmers market. And so Canna Research and Canna H2O went up there, and they met up with Bam. They got the plants. They then brought them back. They had some other stuff they were working with. I think it was Pineapple Express from, I'm not even G13 Labs, I think. But anyway, they made their cross. The dad was involved, and they all worked on it together and have like bred that variety, and like they brought that to the community. It's a unique thing. It's somebody asked about it earlier today and asked about, talking about it, and was quick to point out like that they knew that it was a qualitative trait more so than a quantitative trait. But that's something that's really, really interesting, and I'm glad that we figured out a way to work it in.
Yeah, that's a very good one, good visual example to use when distinguishing between those two, because it's unique for sure.
Yeah, yeah, and also we just grew out a bunch of the holy banana this year, and there are just freaks in that line. Like this was an open pollination; it wasn't holy banana. It's like holy banana crossed to a whole bunch of other stuff from the homie Jos Peros bred. But it some absolutely amazing phenos in there. There was one though that had it was trifoliate from the very beginning, and it maintained its trifoliate structure at least until 6 feet. I stopped
Oh
paying
wow!
attention at that point. Yeah, it
Holy
was
cow!
like super branchy, and one of the branches had a like intense variegation, like yellow, just like
Wow.
albino branch. Basically, there was
Huh.
a little strip of green on part of it, but it was like basically 60% or more, maybe even like 75% yellow branch.
That's weird. I have not run into that yet.
Yeah, it did it all the way through flower. And so, yeah, definitely it's a line that has some unique, it has the ability to throw things up that are just really, really unique.
I'd assume so. I mean, there has to be so many recessive things locked into that particular strain just to get it the way it is. Again, I only put down two. I got two different morphological. One looks a little bit more like the ABC. The other looks like the fern type freak show. The fern type freak show has more of a gassy profile. The one that was more ABC is a lot more fruity. So I did get some differences, but fun and freaks either way. So
Yeah, yeah.
yeah.
So, Kevin McKernan, mentioned him just a minute ago, and Reggie Godino. That's Reggie Godino is Steve Hill. Kevin McKernan is Medicinal Genomics. McKernan et al. In 2019, they did a genomic characterization of a complete terpene synthase gene family. So this is a good paper. It just adds more information. So they identified 55 terpene synthases. They did not identify 55 TPSs, just to be clear. These are the actual synthases. They analyze the genomic content. So the overall number, to my knowledge, that has been identified of TPSs is 35. But there are much more than just 35 terpenes, if that makes sense. There's like hundreds of terpenes.
Yeah, I think over 200 was the number I saw last, and that was a recent study.
Yeah,
But
yeah. So there are tons of terpenes, but they've identified 35 terpene genes, and here they've identified 55 terpene synthases with genomic content. That's not the same as TPSs, as far as I'm aware. Now, this is a different type of paper. So Mudge in 2019 published her dissertation as well as an interesting paper. And this looks at chemometrics and metabolomics. So in it, she basically, the thing, if Booth wasn't able to find genes for terpinolene in some of her earlier work, and so terpinolene's an underrepresented represented terpene in Booth's work. Mudge was able to find some terpinolene expression, some unique factors associated with terpinolene expression. And I quoted part of her thesis because it talks specifically about it. But there was a strong indication of a unique biosynthetic pathway for the synthesis of terpinolene, 9 additional monoterpenes, and their identified cannabinoids. Isolation and characterization of the most prominent cannabinoid identified was THC-AC4 with a butyl side chain. Another correlated cannabinoid was identified as THC-VA. These two cannabinoids are present in higher quantities in the presence of terpinolene, suggesting that breeding for increased terpinolene production switches on the biosynthesis of terpinolene in some traits. So this is something that's very interesting in terms of identifying, well, one, that this seems to be a trait that switches on and off in relation to a particular threshold of cannabinoid production, and two, that terpinolene expression is associated with this high THC production. It's one of those things. I have a friend, his name's Reggie Weedman. He took a bunch of classics down, like all the great cuts, the Urkels, the OGs, all the flavors, took them down to Maui and had seeds and stuff and a lot of good clone-only varieties that he had sent to him. And he came back and he brought all these different flavors, and he would open all these jars of really incredible herb, and he's like, I don't know, man, all roads lead to Jack.
I've heard that. Yep. I've heard that before. Well, it'd be interesting if, we were talking earlier, the terpene synthase genes, if you found the one for terpinolene, and then you found that in the early tests of the vegging plant, then you could almost assume that that plant might have increased cannabinoid production, just because it sounds like from Mudge, the two go hand in hand.
Yeah, and I think that I think really what she was finding is this really extreme expression. And that part of, I would say that sort of the Neville's hazes kind of represent this to a certain extent, because, I mean, some of them are just like disgusting terpinolene intense terps. But then you fucking smoke them, and as much as you may not necessarily like that terpene, it's fucking insanely potent. Like, some of the most racy potent herb that you've ever smoked. And you can see where the potency and terpinolene expression seem to be associated on some level. Now, they don't necessarily, it's not necessarily a one-to-one. Myrcene is by far it's the predominant terpene across all cannabis, but terpinolene is a beast and seems to dominate a large subset of high potency cannabis, and that the other subset of high potency cannabis is dominated by myrcene.
And that's interesting. We could be staring at the answer for that right now with that connection in the paper.
Mm-hmm. Yeah. Okay, so I mentioned this a little bit earlier. This really goes into the specifics of the metabolic pathways that produce terpenes and cannabinoids. So this is the metabolic pathway that leads to sesquiterpenes. This leads to monoterpenes. And right here you'll see that this is the same precursor that is necessary for CBG, which is the precursor for essentially, all terpenes. Excuse me, cannabinoids. So yeah, this is just to demonstrate that there's every one of these has genes regulating them. Every one of these has environmental variables that interact with those genes. All of this is an incredibly complicated set of genetic factors and environmental variables that produce the end result. The one of the things that we'll get to in one of the later papers here, I think it's Grassa, that perhaps selecting for things that increase these different precursors and the amino acids that are produced upstream from the actual cannabinoids will improve overall THC, or, you know, if you're trying to breed for increased potency or increased numbers of a particular cannabinoid, that increasing selection for these things and for these things that produce more of this and more of this will end up producing a higher percentage of this over selecting for more copy number variants of just this, if that makes sense. So then here, this is an older version. Now, granted, I believe this is from Booth, but this shows a linkage map on the chromosomes. So you've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 chromosomes. These different colored numbers are, you know, alphanumerics, and their lines indicate different either terpene synthase or isoprenoid biosynthase or cannabinoid biosynthase genes, and where they map onto the chromosomes. This is a very early model. It's a little bit more refined than the type that you'll see in the early D. Maher papers. Here's a more recent one. I believe, yeah, this was from Livingston in 2020. Again, you can see chromosome 3 here, chromosome 6, chromosome 7. There's total cannabinoids, percentage of THC, percentage of CBG, percentage of CBD, and percentage of CBC, and where they map onto the chromosome. Here is a more in-depth terpene and cannabinoid map. So again, this is something that can now be done very quickly in a single experimental trial. Doing this type of linkage map assembly for a crop years ago would have taken a long time. And so we're able, you know, the fact that we're having our crop of interest come online at a time when we have advanced genomics, we're able to find these assembly or develop these assembly maps much more quickly and theoretically use these, because each one of these is a different marker. And each one of these markers is also part of a linkage group that you can then evaluate their inheritance and their presence in a population. So when you're saying, if you're able to look for one trait, does it give you some sort of indication of the presence of another trait? Not 100%, but this right here, all of these genes are together. And the presence of this gene increases the likelihood of the presence of this gene because they're linked and they're part of a linkage group. So it's not 100%. Like, it's not like, you know, male-female sex kits aren't 100% either, but like just assuming they were a simple binary yes-no or male-female, it's not that simple, but it gives you some degree of indication of relation. There's a lot more work that's gone into cannabinoid expression and inheritance. Early work was primarily done by D. Maher And he proposed a single locus with a codominant allele. So this was the B locus with a BT and a BD allele. This explained the distribution of the 1-2-1, where you have one pure THC chemotype, then you have two mixed chemotypes, and then one pure CBD chemotype in an F2 population when you cross two inbred type 1 and type 3 cannabis lines. This was the first early experiment and explanation for inheritance and trait expression and in cannabis, but this was wrong. But De Maher, I don't want to give the impression that like there wasn't any reason for them to come away with this conclusion. Like the math, this is the simplest, most parsimonious sort of explanation of what they were seeing in the data. And they also, they maintain the possibility for an alternative explanation being a quantitative inheritance as opposed to just a traditional Mendelian with codominant alleles type system. Van Backel in 2011 published the Purple Kush Genome and Transcriptome. This has led to an early hypothesis of two locus model for cannabinoid inheritance, and they also were the first, I believe, to propose that there were non-functional genes encoding for THC and CBDA synthase, and that this was determinative in the outcome of phenotype expression. And then Weebland et al. Confirmed and extended the findings of both authors, which can be kind of counterintuitive, but they were able to extend the findings of Van Backel and the hypothesis of a multiple locus, non-functional, functional allele. And they were also able to confirm just the chemotypic segregation and distribution of De Maher's. So it's one of those things where the overall phenotype data that people were looking at very much indicated a segregating population that was operating on some level on a qualitative inheritance scheme. But, and you know, that was the early studies. They all kind of like, they're all operating and talking in that sort of universe. After the Weebland paper in 2015, shit got way fucking complicated, and we'll get into that. And it's interesting.
Way complicated. I love that.
Yeah,
definitely. I think we're all in agreement there. But sometimes you're only as good as the information you're working with. And so as time goes on, we learn new things, able to test for new things, and yeah, that it progresses the knowledge.
Yeah, no, that's so true. Like, I think the indica sativa thing is such a like product of the 1970s, and it's not that like, you know, you had science-minded people reading the science of the day, and you had the Schultz paper and you had the small paper. That was the available information, you know. And Schultz was way cooler, not gonna lie.
That's awesome.
He may not have been as like scientifically rigorous, but like dude was ahead. He was legit. So this shows, this is again NAPRO. Chimera worked with NAPRO. NAPRO did some research, a lot of research. They did really large, massive testing of cannabis in the commercial, independent testing on their own. They went and bought a bunch of samples from all the different clubs during the 215 days. They brought all these samples back and they tested all these samples to see, you know, THC, the cannabinoid expression, as well as the terpenes. We saw the slide earlier that was from there. And they used this, and they did a lot of the work in terms of doing the genetic information, the environmental information, and the phenotypic expression, and tracking that, and being able to really do the sort of regression analysis that's necessary to track the breeding value, or the breeder's equation that we talked about earlier. And so this was part of that process, and this was really, this data set right here comes from sort of their early R&D, where they were going through and just looking at what was available on the commercial rec market. And really, they're also some of the people that found that the rec market was highly skewed towards myrcene dominant terpenes, and that there was a significant lack of caryophyllene, particularly caryophyllene type 2 varieties, and they ended up producing some varieties. They also got some patents that were very controversial, and they also won some cups at the Emerald Cup, which was also very controversial. But that was the business Biotech Inc., I believe, that was a customer that worked with NAPRO. So they're separate entities, just to be clear. But NAPRO did the scientific research that helped to facilitate the breeding program that ended up producing those results. Okay, so this right here is not a great data set, but it's a data set. It shows these are potency trends from confiscated cannabis samples from 1995 to 2019. This right here marks hemp legalization. So,
Oh wow. Okay, yeah, that's interesting that there is a dip, but hemp, yeah.
Yeah.
Huh. Okay. And you would think though confiscated um goods would actually be somewhat of an accurate reflection of what was going on in the market. It's not the Mississippi University stuff.
It is, and it isn't. I didn't include all of this one shows the clearest trend line, and so I cheated. I could have picked, there were two other images that are much more complicated, and I think part of it is that I can't be entirely sure. I didn't really look at the data set again. It's been a while since I really looked at this, about a year ago or 6 months ago. But the data just looked fucking stupid. It looked like for domestic samples, the trend line for here was down like this, like the reason this line technically is depressed the way that it is because there was this trend line that basically stays stable and static at like below 5%. And I was like, the fuck? Like, nuh-uh.
Who's smoking that?
And I think it goes to like here, actually, technically. And the difference is that they stopped eradicating ditchweed at that point. So I think they were fucking up the data on domestic cannabis by including ditchweed in their sample set. So you have like this here where you have a much higher rate of 3% or under THC varieties. I think part of that is being skewed by ditchweed. But that said, this trend line, when they stop doing that, still shows the same fucking thing, which is a fucking just astronomical curve. Like, yeah, there's a dip, but like here, you know what that dip is? This kind of correlates pretty closely to the people really fucking figured out what CBD was.
Okay.
Like, 2010 to 2012 was like early project CBD time. Like maybe 2008, 2009, the very first samples were tested in the California rec market. But like here you have some leveling off, and then you have an enormous fucking takeoff, which is correlated to fucking, or, you know, associated with the California rec market coming online, it looks like. But basically, the point is that this trend line shows significant increase in THC. We all know that there was significant pressure for selecting THC. We also know that nobody was doing regression analysis, or, you know, very few people were doing regression analysis on large-scale populations, but were instead doing recurrent selection for really, when you talked about what's XD and what's the importance of the XB or whatever in the selection differential in the breeder's equation, just by constantly selecting in the higher upper threshold of THC percentage as a phenotype, usually through a bioassay of people, you know, had smoke and shit, we were able to create this sort of gain in the trait expression. You know, all things being equal and recognizing this is an imprecise data set, you know, this is by no means a actual study where you can make a one-to-one causal claim, but you can see the trend line here and the intense increase in THC percentage. And if you break it down, this data, like the highest THC percentages were actually like, there was some crazy high 30s in the mid 90s that happened. So like, it doesn't just track to where it's like, you know, oh, now everything's over here. Like, you can see where different high THC varieties have been sampled in the population or out of these samples, and very low as well. This all comes from the University of Mississippi's fucking program on tracking potency, which is all, again, kind of what we talked about earlier today at the start, where we were talking about how a lot of this information has been tracked by the cops. And the information that we have is, you know, kind of determined by their interest.
Yeah, not always fully invested and accurate as long as it gets a conviction. But so serves as a guideline, serves as a guide.
Yeah. So this is Hillig and Malberg. They really just point out that there's a qualitative and quantitative dynamic to cannabinoid inheritance or expression, and really the qualitative characterization involves determining a plant's ratio. So the chemotype, that idea of like it's a 1-to-1 mixed chemotype, that's a qualitative characterization, making a characterization of a pure THC or a pure CBD or a pure CBG variety. All of those are chemotypic distinctions that are qualitative. They're isolating the different qualities of the traits that are discrete qualities that they either do or do not have, or, you know, they're either a mixture of the two in the terms of the mixed chemotype, or they're divergent. He also addresses the qualitative nature of the trait. I think it's been this whole idea that D. Maher established, it's kind of, this was old hat. Yeah, Van Beckel and Weeblin updated it and introduced the idea of more of a qualitative or quantitative inheritance. But now here we get to the part two. So cannabinoid expression and inheritance has gotten much more complex as genomic research has developed. And what we've found is there's the work of McKernan and Vergara looking at copy number variants. I've mentioned this a couple of times, but basically this is different genes that are copied multiple times across the genome, and they've been shown in a number of different other species, not talking about cannabinoid genes specifically, but in other species, copy number variants have demonstrated an increase in secondary metabolites. And so that's an area of emphasis. McKernan's looked at it. Vergara's done some work there. Grossa has done some interesting work. There's a very, very interesting findings in that paper that looks at a specific set of QTLs. They also challenge the copy number variant hypothesis. They think that the variation is much more potency is explained by, like I had mentioned earlier, some of those precursor or upstream metabolic pathways. And then the Campbell paper is fucking crazy. Like, this is where shit gets really complicated. This paper looks at, yeah, I'll just wait until I get there. It's wild. Vergara looks at copy number variants, and like I said, looks at how basically every species has these. It's a fast way of acquiring diversity, rapid adaptation, strong selection pressures, and a key component of genetic variation within species. So we have a very adaptive and plastic species that we're dealing with. Cannabis is incredibly adaptive to all sorts of different environments, and it's developed all sorts of different mechanisms for making it so it's adaptive in these different environments. Copy number variants are probably one of those things. It's not a big surprise that cannabinoids, which it's the only plant that produces them, particularly at the numbers or the quantities that it produces them, it's probably got some sort of evolutionary adaptive function. And as selective pressure, i.e. either environmental pressures or human selective pressure, these copy number variants have increased, and there are some studies that indicate that it is a response to human selection. Two major cannabinoids, THC, CBD, are products of three-step biochemical pathway. We saw that earlier. Using whole genome shotgun sequence data for 69 varieties, they found genes encoding for synthase in this pathway varying copy numbers. So that was an early study looking at it. This is really one of like McKernan's work with the Jamaican lion and their genome sequence information and reference genome. They've really done a lot of work at medicinal genomics and have published a number of really, really cool papers. The findings in this one are, one, confirm the idea that copy number variants for THC synthase are present in cannabis, that there's quite a few of them, and that this could explain increased potency or increased THC production. But what they also find, which is very interesting, is that there is a link between, there's a genetic linkage between THC copy number variants and so yeah, here. This identified multiple copy number variants governing cannabinoid expression in 82 genes associated with resistance to, fuck, I wish Matthew was here.
I have no idea what. I wouldn't even know where to begin with that one.
Powdery mildew.
Okay.
Yeah, so this, aka powdery mildew. So results indicate that breeding for plants with low THC concentrations may result in deletion of pathogen resistance genes. A refined genetic map of variation in cannabis can guide more stable and directed breeding efforts for desired chemotypes and pathogen-resistant cultivars. So this is really cool. And the thing that I just skipped over is also cool. So low THC cultivars have a polymorphism every base, every 51 bases, while dispensary grade THC cannabis exhibited variant every 73 bases. So what that means, if you have more variation in low THC, i.e. hemp varieties, than you do in high THC dispensary varieties. So there is less variety here in the dispensary grade varieties than there is in these low THC varieties. And that's because selection pressure has been more intense here, reducing the number of overall base polymorphisms.
Okay, yeah, that makes sense. We went away from those low THC cultivars because they didn't do what the consumers wanted. So who needs them?
Yeah, and here for this, there's been a lot of intensive breeding and selection. And so the stuff that you get at the dispensary has been put under a pretty intensive selection that's resulted in, fewer polymorphisms than you see in varieties with lower THC. So lower THC is typically a sign of less selection pressure, and high THC is a sign of high selection pressure. So these are the things we've been selecting for. These are not. Sorry, jump on in.
Oh, no, you're fine. I was yeah, you said it the way that I meant to say it, but the words did not come out of my mouth.
No, I actually, after I said it, I was like, fuck, I just repeated what he just said. We're all good. So Grassa et al. Had some very interesting findings. This is 2018. I think it's actually 2021, but this was from the preprint. Same paper, either way. This is them questioning the idea that copy number variants of THC genes are responsible for the variation that's being seen. So what they found, or what they say, is none of the 5 separate QTLs that they identified for total cannabinoid content or potency were associated with cannabinoid synthase genes clusters. For example, the strongest QTL for potency, accounting for 17% of variation in cannabinoid content, was located on chromosome 3 rather than chromosome 9. This suggests that traits or genes, gene regulatory elements not linked to cannabinoid synthase gene clusters affect cannabinoid quantity to a greater extent than synthases themselves. Then the next thing, they go on to explain, we might expect gene expressing other metabolic enzymes upstream of THC and CBD in the cannabinoid pathway to be associated with potency, and they identify specific ones. So the hexanate pathway, the methyl thriotol phosphate path, the MEP pathway, and the GPP pathway, and so all of these, they've identified specific ones, and they locate genes in our assemblies and verified their expression. We then compared their physical map position with our genetic map and found 2 candidate gene proximal to potency QTLs. The gene coding for AAE1, the last enzyme of the hexanate pathway, is located at 39.7 centimorgans. So this is, we talked about centimorgans and linkage maps. So on this chromosome, at 39.7 centimorgans on chromosome 3, there is this gene, and it's associated with 17% of the variance in potency, which is quite a bit, relatively speaking. Then, anytime you're talking about the genetic factor controlling a trait, like we saw those heritability numbers,
Yep.
A lot of that is that genetic control for that trait. So this right here controls 17% of the trait that you're seeing expressed phenotypically, which is, even though it doesn't sound like a lot, we're not talking about, there's a very low probability that it could ever reach 100, is kind of what I'm trying to say. The gene coding for this badass HDR, the last enzyme in the MET pathway, is located at 1.61 centimorgans from a QTL on the X chromosome that is associated with 9% potency variance. Now, this is dope. So the X chromosome makes this a sex-linked trait. The fact that this is present on the X chromosome means that if this were on the Y chromosome, if they had found this gene on the Y chromosome, then you would have a male sex-linked trait on the Y chromosome that explain 9% of potency variance, which would be a really, really, really good reason to breed with males. But it's on the female, the X chromosome, which is a good reason to breed with females.
Yeah, that's kind of what I was thinking here is that some sort of indicator that potency typically passes on from a female?
I mean, it's a gene that controls 9% of the potency is connected to the X chromosome. It's on the X.
Okay.
So it's going to be present in an XY. Like in an XY, excuse me, it's not necessarily going to be present. There is always the possibility because of the chromosomal system of the X and the Y. So you're always going to have at least one X present, even with males. However, if you're breeding with two females, you're increasing the propensity that you're going to have a gene that codes for potentially 9% of potency variance. So if you're combining two females, you're probably going to increase the probability that you're going to increase potency as opposed to reduce potency because this sex-linked trait. Now, I can't guarantee, like, this is me, guy, reading a paper and being like, I'm pretty sure logic allows me to deduce this.
Right.
I haven't tested this in any way. I can't say for certain, but I can say that we have some pretty good, like, anecdotal evidence to support this claim, along with the other anecdotal evidence that we have to support the claim regarding light sensitivity and hermaphroditic expression. Like, I mean, I have a fucking really dumb fucking old, Takashi 69 meme where he's testifying and he's saying all your favorite seeds are bag seeds because, like, OG chem cookies, like, they're the vast majority of the gene pool right now, and they came out of like the origin story is accidental mistake fucking crosses and bag seeds.
They are, and that's interesting because people do like them for their potency. It's nothing hits like the real chem dog. But again, bag seed presumably was a herm on a female. Wow, that's an interesting argument. Again, there's always going to be pluses and minuses, but that's another kind of interesting argument for people going the selfing route, as far as maybe trying to increase potency in a particular line. Interesting. Yeah. Okay.
So Weeblen also Weeblen's up in, I believe, Minnesota, University of Minnesota, but he works with Muhammad. I don't want to butcher dude's first name. El Sol, I believe, is his last name. He's the guy that runs the fucking UMISS and has ever since Carlton Turner left or Tucker took off to whatever his name is, went and became Reagan's drug czar. Anyways, I digress. They published a paper. This paper was looking at predictable inheritance of chemotype when crossing a feral, an industrial hemp, and a like drug variety. And they confirmed that there is a QTL that controls the trait. there's at least 5 different chromosomes. It's on at least 5 different chromosomes, but that was just in reference to the Grassa paper that we just looked at. And that yeah, there was something that I thought had been interesting about this paper, but it's not. It's just another one proving the point. Campbell, this paper fucking is bananas. So, unlike previous research, we know non-additive components, so that's epistasis, of cannabinoid inheritance. Concentration of THC is a polygenic trait, 3 or 4 genetic factors. Both additive and dominance, I can't remember what CGE, but it's genetic. It's something, yeah, I can't remember what CGE stands for. But it best explains THC expression patterns. In contrast, cytoplasmic genomes and additive genes may influence CBD concentration. Maternal additive effects and additive genetic effects apparently influence CBC expression. Conclusion: cannabis inheritance is more complex than previously associated. Among other genetic effects, cytogenetic and maternal contributions may be undervalued influences on cannabinoid ratios and concentrations. So this is a whole nother level of variability that we're not just dealing with the nuclear genome, you're also dealing with the solely maternal influence here of cytoplasmic genomes. And yeah, so that this also adds to the Grasses paper and provides additional explanation for how the influence of additive maternal effects could influence at least CBC expression. But when you go into the paper, they go pretty deep. And I gotta say, like, I read this, I think, yesterday, and it was kind of like a mind blower in terms of adding a whole nother level of complexity to what, I mean, like, I'll be honest, like, this part of, I thought I understood a lot of this stuff. And, like, you know, as cool as this passage right here about the presence of this gene on the X chromosome was,
Right.
like, I was like, okay, yeah, this is within, like, what I was expecting. I wasn't expecting to also consider, okay, we also have to look at the cytogenetic effects. We have to look at essentially what is the CP, the chloroplast, and the mitochondrial DNA, and the effect that that's adding and contributing to this trait expression. And, you know, it really is layers on layers on layers of complexity. And when you start, like, peeling the onion back and really looking at this stuff, it's so incredible that people were able to, like, people were able to, like, make food crops and, like, make them do what they did with those, and that, like, we can eat. But then, like, with this plant, like, just that graph of, like, potency in cannabis, it's like, given how complex this shit is, wow. And, like,
Right.
perhaps some of the stuff that we were doing all along that we didn't necessarily, you know, we didn't have all of the tools to discern what we were doing. We were just simply smoking it and saying, yeah, that works. Yeah, I like that. We were doing the things that these people are now elucidating, and, like, they're illuminating what we've done and the work that cannabis breeders have done for generations, and are explaining to us shit that we did without necessarily having the scientific ability to characterize and articulate what we were doing.
Yeah, we had almost just had that third sense of doing it. And it was, you know, it was rapid or intense farming as well. You know, sometimes it takes a while to develop the new corn variety or the new apple variety or whatnot. But this was pretty intensive farming. You know, every run you're pulling that, you know, what you perceived as the higher THC or, you know, the better stone, you're pulling that one out and getting rid of the, you know, what may be CBD. And I'm curious too on that last study, I'll have to look at that. I'd be curious if for the maternal contributions may be undervalued in the effects of cannabinoid ratios. I'm curious if they cited the previous paper to get that information in there, or if they came to the conclusion through their own means.
Yeah, no, it looked like it was on their own. They made a lot of references to Dima Hare and to other authors, but it seemed like they were like a lot of this the stuff about like non-additive and additive components seemed to be really unique to their particular method of analyzing this issue. And like there's this paper and the Mudge paper and a couple others that have looked at different omics that I think are some of the things that are really, they're changing the way that we understand, and they're really like changing the level of analysis. Like they're taking individual like trichomes and using that as the cell that they're then doing the genetic. They're like literally trying to isolate just the transcript information that's going on in the trichome cell and understanding what's going on as the RNA is translating the DNA to make it so that it produces the proteins and the enzymes that are producing the end results that we know as, you know, THCA or different terpenes. And the fact that they're getting that level of resolution, and like that we have a plant where you really have to do that level of analysis. Like looking at the leaf tissue isn't all that informative if you're really wanting to look at, you know, the specificity of how and what is going on at a genetic level inside the trichome. And if that's really what the fuck it's all about, which is so much of the ball game in our plant, not to say that the leaves aren't important, but like you got to have that level of discernment. And also some of that terpene analysis, it's I guess the roots have some very unique and specific terpene genes or expression in root cells. So this picture is just another indication or another demonstration of the chromosomes and how they're being, the linkage maps are being drawn out on them. And again, I tried to explain this earlier. You've got your autosome, your sex chromosomes, and yeah, and it's a diploid plant. So each one of those is got a double chromatid. This one shows that right here. Sorry, you can jump in anytime, Chad.
Yeah, no, you're good. I'm sorry, I just unmuted there for a second, but yeah, we're all good. Keep going. Thanks.
Yeah, no, I only know what's going on through that sound. But so this right here, this is another representation of kind of what we just saw. Here you have the genetic map. There is, where is it in? So basically this zone right here shows that genes responsible for chemotype on chromosome 9 are under selection in marijuana populations and have been the targets for introgression by breeders. So we've been doing heavy selection. And when they do these sort of genetic tests, they can see, okay, this area is under intensive selection, and it's upregulating for the expression of those genes because breeders are actually trying to select for these. And part of the way they're able to do that is by looking at that those base pairs. So the differential in base pairs is part of the analysis there. And here it's very clear that on this particular chromosome, so chromosome 9, they're able to see a particular segment of the chromosome where where selection has been intensive for cannabinoid synthase. So yeah, we covered cannabinoids, we've covered terps, flowering time, some interesting papers are coming out on that. Schilling really just says, hey, it's possible to do this. The PAN 2021 paper is very interesting. This talks about Constans-like gene family. This is one of those gene families that's found in a lot of different crop species. It's been characterized in pretty much every other crop species, and because of that, they're able to find similar analogs in cannabis. And this paper does some of that work. And then the name Feel et al. 2021, we looked at this. This is the paper that did some of the work on specific broad sense heritability in cannabis for traits like yield and dry flower yield and height and a number of different traits. Like I said, Schilling said this shit's possible. Oh, but yeah. And they also explained like how to do it. So it remains to be seen how complex traits will be determined in cannabis, but taking inspiration from the successful elucidation of complex traits in maize may motivate the formation of sophisticated multi-parent mapping populations, such as nested associated mapping or multi-parent advanced generation intercross magic populations. So these are different breeding programs or populations that you can create through breeding that will elucidate these traits. And this is how you would create these populations, and then you would track the traits, in this instance, flower time, over these different populations to be able to infer, well, one, to determine on this level genomically the actual markers associated with the trait. Absent marker-assisted breeding, you could do it through inference using the methods that we talked about earlier. So Pan et al. 2021 did some of this work, and they were able to demonstrate that flowering period of plants is a complex quantitative trait. Lots of shit going on, lots of environmental factors that also influence. So there's a lot of variability here between genotypes and environments. Studies have shown that constant-like COL is an important regulator of plant response to photoperiods and is a core element in the regulation of plant flowering. Unfortunately, I didn't cite the more relevant part to our interest, but I believe they found a specific number of genes that are associated with it and characterize those genes. And now we have the ability to start to build out, much like the terpene synthase genes, a whole architecture. And there's really good images. I couldn't really fit any because they were too big on the terps, but there's good images showing the phylogeny and how all of these different genes are not just like associated in cannabis, but like with all other species that also have these same analog genes. So name, feel, this is the, this describes the paper that they did, and this is a very interesting paper. It's really worth going into, and you know, to the extent that you can apply some of these insights, it's something that could actually improve one, standardization of data collection and utilization of that data collection. So a diverse population of 121 genotypes of high THC and balanced THC CBD ratio was cultivated under a controlled environment, and 13 parameters were measured. They found floral bud weight, dry weight was positively associated with plant height and stem diameter, but not with days to maturation. So there's a negative correlation there. The heritability of both plant height and days to maturation was relatively high, but plant height decreased during vegetative growth. And basically, you do this shit, it could help improve. Oh yeah. To advance breeding efficiency and prediction equation for forecasting floral bud dry weight was generated driven by parameters that can be detected during vegetative growth phase only. So essentially, they're claiming that they created an equation and a prediction model that just using vegetative growth information can help you predict your yield and different outcomes. Yeah.
And does that get into the chemotype as well, or is that just morphological aspects?
They really primarily look at morphological aspects. They're basically looking at how to optimize yields. So
Awesome.
this is like a really good paper for if that's your jam and you really want to like do data optimization to increase productivity and figure out ways of working on predictive equations that help you select genotypes that are going to be particularly productive. This is the paper you want to read. Like this shit, it slaps. And like I said, we've seen this already. This is the broad sense heritability of different traits. And yeah, this is the only time or the first paper I've seen that actually does that in cannabis, and that's the end of our jam.
Damn.
Yeah, we
All
made
right.
it.
Well, yeah, you tell this is pretty cool. This is coming up this week. Are you going to be one of the speakers for the virtual aquaponics?
I'm not, but my homies, like so many homies on this list, great people, and
Oh
just
yeah.
Aquaponics is a really great person who's reached out to me, and I hope to be on the Growing with Fishes podcast here soon. And
Nice.
just it's one of those things where I appreciate everything he's done, and so many people on this panel are just I can't like I cannot encourage people enough to like. Breeder Steve, who he was mentioned earlier,
Yep.
he's brilliant. He is an incredibly nice human being, which is also nice. That there are people who reach his level of accomplishment who aren't friendly to random folks, and he is one of the nicest people around.
That's awesome, Near. He's a funny guy too. I've never met him myself or had a one-on-one conversation, but I've seen him in a lot of interviews, and he he's a pretty funny guy too. So that's awesome. But yeah, it's an all-star lineup of extremely knowledgeable people. It's free, which is huge. And if you thought our 4 hour, 5 hour, sometimes 6 hour shows were long, strap on the diapers for this because you're not going anywhere. You can be full for the entire weekend. So it's an awesome thing that they've all come together. And yeah, Potentonics kind of being the rig leader there.
Yeah, totally. So, shout out Dragonfly, shout out Caleb Copyleft Cultivars. Great
Yes.
work is being done here. Definitely check out Copyleft Cultivars. They're doing work on IP with genetics and really trying to develop a framework that will help protect sort of a open source culture within the cannabis genetics community. So great. Can't wait to hear what he has to say. Chris Trump, KNF, That's what's up. Heavy days, always great. You know, the podcast, listen to that shit. It's
Oh
like
yeah.
Wendy, you know, she's KNFing, going hard, doing big stuff. It's beautiful to see. Matthew Gates, he would have known how to pronounce every scientific term I had trouble with tonight, and it's just brilliant.
I agree there. One person I would never want to play Scrabble with, but I'd enjoy a cup of coffee.
Dude, it's done. And you know why? It's because he knows all those small words too.
Yep.
Yeah.
He's awesome. But yeah, and then
Also,
you mentioned Breeder Steve is kicking it off, looks like Sunday morning.
no, bright and early. So that's one, like, if you are still listening to this, like 8 a.m. fucking PST, get on that shit. It's gonna fucking slap. Dutch Blooms, homie, he's a good dude. Kevin McKernan, also Dutch Blooms. Like, you know, you catch him on the round tables.
Yep. He's next Friday, 1 week from today, we're gonna have Dutch Blooms on here. We're gonna be talking about the regenerative conferences.
That's dope. Yeah, no, he totally spearheaded that and like helped get that thing rolling. And honestly, like, you know, incredible, like, much props to everybody involved in that, because there's not a whole lot of spaces that still feel like they're in and of the community. And
Right.
I rolled up to the Mendo one, you know, I think it was 19 now or 20, you know, it was before lockdown, and we got down, and you know, good people, and it was really good. It's good to see everyone there. Mr. Bob Hemphill, all those folks who rolled through to the Mendo scene, and Kevin McKernan, he's been cited like dozens of times here tonight because his work with Genomics is incredible. Way back, there's a YouTube video link. Check out that video. It's really incredible, his work. Clackamas Coot. Anybody I haven't mentioned, I mean, no disrespect, I just don't know these people personally, but like, I'm sure they're incredible speakers given who they're speaking with. Clackamas Coot has literally built like the fucking damn near every organic cannabis nutrient line company industry is like riding on that guy's back on so many levels. So,
Yeah.
props to Coot. And yeah, so I just wanted to give a shout out to my homies and to everybody who's putting this on and making it a thing. Jordan River, fuck yeah, moderating on the craft grower panel. That's going to be legit as well. So it's like literally, like I just went through 48 hours or whatever. I can't count, you know, the math
Yeah.
on the thing, but you know, banging.
It's gonna be insane too. And I see the for the whoops, wrong camera angle. But for the home growers panel, Scotty Reel. Scotty Reel is one of the personalities from the Do Grow show. Uh,
Right.
he's a freaking riot. I've been on their show a couple times and talked with him, and that guy's a riot. So that'll be fun. That'll be interesting to catch too. I know that the home grower panel there right before the craft panel. But yeah, that's man. And the cool thing and a huge plus about that is we're gonna have, or it's gonna be available for replay on YouTube, because there really are a lot of people I'd love to catch there, but I don't have that much time in the day. So I know I'm gonna catch a lot on the replay.
Yeah, totally.
Yeah.
But no, so that you know, check that out, and thank you everyone for listening. If there's any questions or anything, like, you know,
Yeah, toss them up. Toss them up here at the end if you guys have any. I know I want to send a big shout out and a big thank you to Mr. Toad. Mr. Toad's been in chat all night. He's been holding it down. He's been offering great information to everybody. So it's like it's the third panelist on the show this evening. So shout out to you, brother. Thank you.
thank you.
Appreciate
No, I couldn't ask for anybody better to, like, you know, anything I can cover with the interface of chat. Like, I know you hold it down.
Yep, he's holding it down. We got Ian down here. So thanks for another epic show, SunGrown. Definitely putting out some epic information, man. And again, I'm I'm glad it's available for replay too, because I've got my notes. I'm writing along. Woo, there we go. I'm writing along as we go. And I'm picking stuff up each time. This is awesome, man. I really appreciate it. And I know there's other people out there that are getting the same benefit out of it. And you're an ace at what you do as far as being able to get a message across. It's not very easy sometimes when you're dealing with complex subjects, but I feel that you put it across in a good way, and you give the references of where we can go explore more. So I always appreciate that too, because
Yeah.
you can't cover it all, and that's a hard thing as kind of an educator or a person talking about these things. Is sometimes you want to qualify everything that you say, but if you do that, you spin your wheels sometimes, I guess, so it is a skill.
No, totally. It's it's one of those things where statistics and footnotes it can be pretty fraught. But I feel like all this information is like the slideshow. Same thing as last time. I'll put it up in like the folder. All the source material, I'll put all that to. I mean, it's all in there, but like it's all over the fucking place. So like, if you already have the file folder that I've like reference and share with people, like you don't know what I'm talking about a Google Drive with a lot of information that I use to make all of these presentations,
Yes.
and it makes it so I can do it quickly because it's kind of organized, but sometimes organization defeats the purpose of organization, and it becomes like I've created too many folders, and now I can't find where, like, yeah. There's one paper. I'm looking for one paper, and I just fucking cannot find it because it explains the phenomena that we talked about of genetic drift.
You mentioned that earlier, yes.
Yeah, I don't know where it is. It's a great paper. I posted it on Instagram once. I can't find it. It's gone. But like that paper is a very interesting paper, and it talks about that variation in like genetic variation in clone plants, and like also the difference between taking the clone at the top of the plant, the middle of the plant,
Okay.
or the bottom of the plant, and like the amount of mutation that somatic mutations that build up. Yeah, wild.
That would be interesting because a lot of times that conversation of top, middle, bottom for clones is framed around the hormones at them to make it root faster.
Yeah.
That's a lot of times what the conversation or like the practical application is, but mutations. Interesting. Yeah, I'd love to find that paper. I'd love to have a conversation even just specifically about that, because genetic drift, genetic mutation, um, talked about for as long as I can remember. It used to be an absolutely happens, and then it turned into no, it doesn't really happen. But if your plant gets sick, it's going to remember that, and that's how things degrade. And this kind of sounds like it's adding a new plot twist to it.
Yeah, totally.
So
And
that would be interesting.
the best part of the plot twist is like, people have talked about it for so long, and we've used the term genetic drift, which is not the term. But that was, like it's just
It's the tools we had.
having a phenomena,
Right.
and like trying to be like, well, gotta be the genes are changing or some shit. So I understand why people said it. People sometimes would be shitty and laugh and go, "Oh, that's not what that means." But like, it turns out that the thing that a lot of times people have been poo-pooed for mentioning the idea, because in theory a clone is a clone is a clone is a clone. Actually, there is some genetic variance there. And being able to discern that and talk about it and have the right terms so that when we go to a scientist and say, hey, blah, 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 is this possible?
Right.
They may not know and maybe like, blah, 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 that's not a thing. But if they're conversant in the subject matter, they'll be like, oh yeah, we see that phenomena here, and like, this is kind of what's going on in other crops, because typically
Right.
that's their area of expertise. And So it's just it was really it was one of those things where that paper in particular was like, holy shit. This thing we've been missing for fucking 30 years of talking about this phenomena where there's actually a word for it. And I can't fucking
Right.
for the life of me.
It'll come to you hopefully tomorrow afternoon, and you'll be like, ah, all right, April this year, got to go back. But yeah, as a person who goes clone to clone to clone because I don't have the plant count or the room, um, yeah, totally fascinated. I'd love to find that, love to read it, love to discuss it. Maybe we could do a study group or something about that one.
Definitely. Yeah.
That'd be awesome.
Yeah.
Well,
You
cool,
know,
man.
I really appreciate this, and appreciate everyone for taking it with us. I managed to smoke basically all 5.
Good job. It's a thorough job. Congratulations, man. And again, I truly appreciate your time and your effort for putting this together and coming and talking to us and, kind of educating us all, not only tonight, but really throughout the week on Future Cannabis Project and FCPO too. So may big buds be bountiful for you, brother. I had to throw another B in there.
Likewise, yeah, no, good times. Thank you so much for everything you do to hold me
Hey.
down on.
Awesome, man. My pleasure. And with that, actually, this whole time I've been flashing, I do have your Instagram kind of going up. Is that the best place to find you, or is there anything else that you'd like to get out there, shout out or anything like that?
Yeah, I mean, I'm an IG. I do write. It had been a weekly, but that pace just wasn't. I wasn't producing as well as I liked for the beard grows on the
Mhm.
weekly pace, so I slowed it down. Um, I also write for Skunk Magazine, so shout out to Beard Bros, shout out to Skunk Magazine, um, shout out to Peter and Daga, Daga Gardens now, Daga Dot Garden.
Yeah,
Uh,
Daga Cars. What did you say before that? I thought you said Bieber.
oh, Peter.
Oh, Peter. Okay.
Peter,
God, I thought you said Bieber.
Peter.
I'm like, where's he going with his shout outs, man?
Shout out to Bieber.
Britney Spears, she's free today. Shout out.
Shouts to my peeps. But no, Peter with Gaga Doc Gardens, and thank you
Yes.
for everything you're doing there, man. And yeah, check out all the seeds and all the products on there. The genetics, like J2 Can is on there. Like, I don't know if y'all know him, but if you don't, like, check his gear out. Uh, Lots of very cool old school genetics coming from him. He's been on some recent podcasts. If you can listen to him talk, it's definitely worth it. And I think it's Bird Seed Preservation is the seed company, but he goes by J2Can on the gram. But yeah, and Jimmy Myers, uh, he he's worked with the same ADHD 13 hack plan as me. We share a lot of genetics. He's somebody who just, I mean, if he ever had time to get on and do an IPM podcast or episode, like,
Be a good.
yeah, like him and Matthew Gates are like
Yeah.
all-star team there. But yeah, so there's so many cool people that are involved in all this. Toad holding it down, like everything. All y'all doing big shit, and I appreciate you. And it's really good to see everybody, like, just all the breeders on there, and also all the different, like, people populating the space and bring home growing networks and different people together so that we're figuring out ways to do sort of collaboratively the shit that we talked about in this during the presentation.
Yeah, it's a good community. It's a good collaborative effort too, and it doesn't really matter age, but it's like you're able to teach people that are new coming into the space, whether they're 45 or 25, but teaching them this information and kind of some of the the ways, uh, it hopefully is going to lead to a better future for the community that we want to see. So, you know, you gotta lay out a map.
Yeah,
So
yeah. And also, you know, shout out to everybody who did it before, you know, like all the people who have, because some of this is reiterative. Like we're going through another process where there was like folks like Old Timer, folks like, you know, Cheap Six Turtles. There was a, yeah, no, if you go back into old Sensi tips and like old books, like the earlier magazines, a lot of the breeding information was coming from him. Like there were cats that were like, that have laid all this foundation out before. And, you know, every 5, 10, 20 years, it's like we have to go through that cycle again because there's new people who haven't necessarily, who weren't there, who didn't hear it from those folks. But like, you know, so it's just, I appreciate everyone who's been part of that process. And I've learned so much from all of the different people who've already like sort of trodden this path that it's definitely helps for them.
yep, the OGs, you know, the Frenchy cannolis of the world who left this space a better place even after they're gone. So shout out to the knowledge, RIP Frenchy. And I guess that'll wrap it up for this evening. I am Chad.Westport on Instagram. You can find me chadwestport.com, and you can find me all over here on Future Cannabis Project FCP02. Deuces, part two is done. So I appreciate it, man. We'll say peace out. The deuce out.